Yes, okay. Anyway, so good evening all and esteemed colleagues, Professor Essen, Dr. Jital, Keval, Suresh, Dr. Uma. So welcome all of you from the faculty. And the first talk would be on longitudinal monitoring strategies and heart transplant and update. This is from Dr. Jasmeet Kaur. She is the head of the histocompatibility and transplant immunology services in Lalpath Labs. So Lalpath is one of the foremost laboratories, the largest network in India. And they are cover almost every geography from India is a very large country, from Kashmir till Kanyakumari and east to west. So I think they have a very huge kind of footprints. So I'll now invite Dr. Jasmeet for a talk.
So, yeah, am I audible? Is that audible?
Yep.
Yeah, okay. So thank you, the Indian Society of Heart Lung for letting me be here and share my experience, and also to listen to everybody and get to know about what's happening in the field of heart transplantation and lung as well. So I plan to speak a little bit on monitoring strategies, a little bit of what's happening, a little bit of what we are going to look at in the in India in the near future. I go back to what's been partially covered earlier, and that is the HLA system. So the whole point of HLA system is to recognize the self from the non-self, which is actually what determines the acceptance or rejection of foreign tissue. And this is applicable across all transplants. So the polymorphism of the human genome is where the challenge in transplant lies to a huge extent. Now, sensitization to HLA antigens can happen on exposure to a large number of non-self MHC. Of course, blood transfusion, the most common, is what we see. And the last two that I've enumerated here are the ones which are more specific to cardiac and lung transplant. With the detection of antibodies came the evolution of DSA. And no doubt the lymphocyte cross-match was the first test that was looked at. And this was with the landmark study of Patel and Thoracic that we became aware of this particular test. And with the evolution of DSA came the revolution when we developed newer techniques for molecular HLA typing, which is something Dr. Uma brought out yesterday. Following that, there was multiplexing of the HLA antibody identification, which has led to where we are actually today. The tools available to us today for typing and antibody monitoring are the ones which actually guide the strategies we are to a huge extent following in risk stratification. There was the CDC, which are the cell-based assays and the modification of CDC, and then a variety of solid phase assays. And now where we stand is at the NGS. We are also looking at the Luminix and SAB with the C1q assays, the antibody IgG subclasses, the donor-derived cell-free DNA, and beyond that, the gene expression profiling. Now, just a little table here to remind us that the basic advantage of the CDC is that it's a functional assay, which is a great feature. Whereas the other tests that we have available, the virtual assays, are those which do not give us functional realities there. So we have benefits and we have limitations of these solid phase assays. They have no doubt increased the sensitivity and the precision, but they have limitations and there are false positives. So as we see the single antigen bead today, it currently provides us at best a semi-quantitative measurement of the antibody strength. So what is it we expect in a solid organ transplant from the solid phase detection? In the pre-transplant, one would hope for identification of donor-specific antibody, quantification of donor, quantify the donor-specific antibodies and predict the cross matches. In the post-transplant, one would actually look to be able to correlate episodes of antibody-mediated rejection or graft loss and to correlate desensitization and the impact of treating antibodies. The pre-transplant challenge here therefore lies in determining the unacceptable HLA antigen mismatches, which is what the single antigen bead does with the help of what we call the virtual cross match, the risk assessment pre-transplantation, determination of the patient's antibody signature, and of course, they do help in helping us transplant the highly sensitized patient. But what is it in the post-transplant that is the challenge? We know immune suppression cannot be too little, cannot be too much. But what we have as of today is a lifelong immune suppression in solid organ transplant, and the drug regimes are by and large one size fits all. But what one is looking at is an immunological risk assessment, which actually is the key to individualized immune suppression post-transplantation. A dynamic process of ABMR in explanted heart allograft was seen, and what Dr. Lupe in her study has highlighted, that ABMR is an indolent process leading gradually to a chronic antibody-mediated injury and late allograft failure. And what one has here is a large expanse of various histological findings which one sees in ABMR. What we really need to understand is that ABMR is guided by antibodies. The antibody concentration itself determines the activation of the pathways that lead to these lesions, whether there is antibody activation through the Fc receptors or the C1q binding or the HLA cross-linking. It is the mechanism of activation which determines the mechanism of the recruitment of cells and hence the type of histological manifestation one sees eventually. So it is not only the concentration, but the subclasses. And we know now that the IgG subclasses, which are from class 1 to 4, of these, the G1, IgG1, and G3 are the ones which are most effective in complement binding. And we know complement is very important in antibody-mediated rejection. Talking about the DSA, the first thing we talk about is the MFI. And DSA, or the risk of ABMR, is increased with the concomitant rise in DSA-specific MFI. But there is this MFI conundrum. A large number of factors affect the DSA to cause organ rejection. And there are situations when a higher DSA wouldn't cause a rejection, and a lower level or lower MFI of DSA would actually lead to an AMR. So looking at this particular study, where they looked at the ABMR in pre-transplant DSA, which was the DSA positive, cross-match positive outcome was worse as compared to those who were negative for both. And then there was those who developed de novo DSA, and that was again worse as far as outcome for ABMR was concerned. And then there was de novo DSA, which if present was not only bad for ABMR development, but also a cell-mediated rejection. So a risk stratification to prioritize the unacceptable antigens would be to look at the single antigen bead, not just plain single antigen bead, which I will quantify in my next few slides, but look at various dilutions of single antigen bead, look at the C1q binding capacity of the single antigen bead, and of course, the CDC cross-match. Now, MFI per se does not reflect antibody concentration, and the strongest antibody being the one that has the highest MFI at the highest dilution. And though Anand Tembur carried out the study in kidney, but it's as relevant in all solid organ transplants, when you looked at they looked at the MFI in the neat serum and at various dilutions, and that went up to 1 in 1024, the highest MFIs were not always in the neat serum. So this led to the concept of looking for higher MFI probably in more diluted serum. The C1q, we know in heart transplant, it defines antibodies capable of binding the first component of the complement cascade. But we also know that there is a lack of correlation between the MFI, which is the antibody binding strength, and the antibody's ability to bind complement. So C1q becomes a sensitive and a specific test to perform in heart transplant, particularly in the first month after transplant. There are no doubt other tests available which can look at the complement cascade and binding in the complement cascade, and that is predominantly C3d and C4d binding. Looking at the impact of C1q in heart transplant, the presence of C1q fixing antibodies also showed a trend towards a higher incidence of ABMR. But this is a finding which is also seen in the kidney, where DSA positive, C1q positive were both responsible for a worse outcome in kidney transplant. So we therefore looking at a contemporary multidimensional assessment of circulating donor-specific anti-HLA antibodies. SAB testing for undiluted serum is not the only test we need to perform. We need to provide clinically relevant information with respect to prediction of allograft injury and loss. One really needs to look at the C1q, binding capacity, and probably also the IgG subclasses. So clinical decision-making now is beyond just antibody detection in both the pre-transplant, where it would be important in organ allocation, risk stratification, as well as in the post-transplant setting. A decision-making algorithm which has been put forward is DSA detection, along with clinical symptoms. If present, go ahead and do an endomyocardial biopsy. If not present, do a C1q. If positive, again, a biopsy. If negative, probably monitor more stringently and watch. So looking at the consensus guidelines of the INSHLT, the important features here are they've recommended post-transplant DSA monitoring at 1, 3, 6, and 12 months. But what is important also is that DSA that remains at higher dilutions, C1q DSA antibodies, DSA that persists, and DSA arising late after transplant has been associated with worse outcome. So those are the recommendations they have out there. And if you look at the clinical significance, and I think somebody also did mention the non-HLA antibodies, the MICA antibodies as well as the AT1R antibodies are the ones which are being studied, but they do not have a definite role, though they have been implicated in a large number of antibody-mediated rejections. So what I really want to focus today is, and what we talked a lot about yesterday, is the new test on the block. And surely for India, it's definitely the new one on the block, and that's the donor-derived cell-free DNA. I just want to introduce some to the concept here. And once you have a solid organ transplant, if there is no active rejection, one would get a very small percentage of donor cells, DNA in circulation. But any form of injury or rejection would raise the percentage of this. And we look at the current transplant surveillance options that are there, they're limited. There's the endomyocardial biopsy. There's a lot of inter-observer variability, not to talk about the sampling size, the area which has been sampled. There is the gene expression assay, which has a high negative predictive value, but it is again a test which is not designed to detect ABMR. Of course, there's DSA and the clinical evidence of graft dysfunction, which is no doubt the most important. So I'm going to focus mainly on the donor-derived cell-free DNA and to some extent also compare it with the gene expression profiling. Now, these two techniques are different. If you look at the gene expression profiling, we are actually evaluating the recipient immune activity. And the table below gives the genes in this particular test, which in USA is promoted as AlloMap. They look at the lymphocyte activation, cell migration, T cell priming, etc. But again, the issue here is it is not designed for AMR. But when you look at the donor-derived cell-free DNA, this test gives us the level of graft injury. And therefore, it is able to pick up ABMR or any form of graft injury. But together, the gene expression profiling and donor-derived cell-free DNA are powerful, non-invasive tools for heart transplant surveillance. And this picture was also projected yesterday, but it just gives very beautifully a comparison. When you look at the gene expression profiling, it gives you immune quiescence, ACR, but it does not detect AMR. When you look at the donor-derived cell-free DNA, it is ACR as well as ABMR. And when you combine the two, no doubt the results are best as far as information to the clinician is concerned. So there's the study with Dr. Kush, where they looked at 2,164 samples, out of which 18 were antibody-mediated and 17 were ACR. So what we see here is that a part of the results of this study were that the donor-derived cell-free DNA was able to detect AMR significantly in these patients. Similarly, the donor-derived cell-free DNA was able to detect ACR as well as ABMR in these individuals. So when you look at the area under curve, the gene expression profiling, area under curve is only about 67.7%. For donor-derived, it is 76.7%. Of course, when the two tests were combined, the score was highest at 80.9%. Another point I want to share here is that the donor-derived cell-free DNA thresholds are different for different organs. And I put the kidney here, and kidney is anything above 1%. In heart, the injury is about 0.3%. And in the lungs, it is 1.5%. Now, why is this little difference there? And the explanation that has been put forward is that this difference reflects a difference in the rate of cell turnover within these allografts. Yes. You probably asked me, the heart is very low. Is it detectable? Yes, the sensitivity of donor-derived cell-free DNA is such that it would detect low levels. So if you look at the timeline for a molecular window for clinical alteration, a normal allograft, and then there is the transcriptomic alterations, which are then followed by morphological alterations and then the clinical alterations. The first window would be picked up, the test that would pick up is the gene profiling and the donor-derived cell-free DNA. The aim would be to pick up the molecular changes as soon as possible to be able to reverse the progression of the injury to the organ. Morphological changes, once they happen and move on to clinical alterations, there could be ABMR, ACR, or mixed when the levels of the test are high. The HEARTS has put forward a duration of testing for donor-derived cell-free DNA. The test should not be done 14 days following transplantation. Can be done after 14 days. The recommendations are to do a monthly testing for the first year, quarterly for the next 2 years, and then biannually for the 4th, 5th years and beyond. The rationale for this is that why would you want to test more frequently in the first year of transplantation? Because that's the time when patients are being weaned off their immune suppression, their steroids, TAC levels are being monitored, and therefore the rejection episodes do come up during this period. Also, the test can be combined, or actually should be combined, with DSA screening at different time points. And that's where a multi-dimensional approach in monitoring these patients becomes significant. So I want to sum up with saying that donor-derived cell-free DNA is an efficient surveillance test. It can be measured in plasma. So the issues with doing a biopsy, a protocol biopsy, or four-course biopsy can be avoided because it's a procedure which is not without its complications. Negative results are strongly predictive of no active injury. The test can be used to evaluate both ABMR, ACR, and its levels will decrease following treatment, which is why the test can also be used for monitoring. And with that, I want to thank you all for giving me a patient hearing. And those are the lovely mountains of Ladakh and such a typical picture from Leh. Thank you.
Thank you, Dr. Jasmine, for a very wonderful presentation. Very nicely put out, articulate. Thanks. Now, our next speaker needs no introduction. He is one of the giants in the field of molecular studies related to organ transplant and one of the originators of the molecular microscopy diagnostic system, MMDX. So, Dr. Philip Halloran is one of the best names in this field. He's professor, Department of Medicine, Alberta Transplant Applied Genomics Center from Canada. So, Dr. Halloran, hope you are able to hear us. And
yes, I can hear you. Can you hear me?
so, please, everyone is eagerly waiting, sir, for your talk on MMDX system in solid organ transplant. Over to you, Dr.
Okay.
Halloran.
Okay, can you see my screen?
Yes.
Yes.
Yes, are we all good? So if our technology is working, I'd like to talk to you about the microscope project, how we have spent the last 40 years measuring messenger RNA in tissues and discovering the molecular changes of rejection and injury. And I'll focus on the heart and myocardial biopsy, the lung transplant transbronchial biopsy, and the lung transplant mucosa biopsy. These are my disclosures. So the unmet need here is, as a clinician, I've always felt we need to understand the disease states. We need to understand them at the molecular and the gene level. And then we need to translate that into precise measurements in the tissue and use that to improve care. I think that's what our goal always is. So the key features of assessing human tissue are precision of the measurements. Is it reproducible? Representation. Does the tissue actually represent the organ? Accuracy. Does it reflect the true disease state? And that means you must always be going back to the patients and checking to see if this explains the patient's problem. It needs to be standardized. Is it the same in Chennai as it is in Edmonton? And personalized. Does it retain the uniqueness of each patient? If you can't measure it, you can't change it. So we need quantitative, precise measurements. That's the starting point. And many of our conventional clinical systems, and I'm including chest x-rays, clinical assessments, histology, they tend to be qualitative or semi-quantitative. They're not measurements. And we tend to defer to eminent experts, and the experts often disagree. So the truth about the disease is what we're trying to discover and assess in each patient. It's never completely known and never will be completely known. It's always going to continue to be a discovery. Our conventional assessments are one estimate, but now we can make molecular measurements and machine learning algorithms to assign a new assessment. The advantage in machine learning is that we can use 100 different estimates, like asking 100 different experts, and we call this an ensemble approach. Now, our goal in the MMDX project is not to agree with histology, but to discover the truth and then see how it agrees, and then look at both platforms and make both platforms better iteratively. This is not a competition. So our strategy for discovering the disease states is to use genome-wide platforms. We use microarrays. We also use RNA-seq. We use supervised approaches, which are based on relationships to existing knowledge, and unsupervised, which are basically the data structure. Then we create diagnostic applications to look at new biopsies, and then iteratively we go back and forth with the patient and bring the new knowledge back iteratively to the diagnostic platform on the genome-wide platform, and then we can copy it onto new platforms using portions of that knowledge in simpler ways. So the microscope diagnostic system is a central diagnostic system that uses microarrays to measure transcript changes with high precision, 99% precision, and compare it to a biopsy, each new biopsy to the reference set using ensembles of 1,000 predefined machine learning algorithms, of 100 predefined machine learning algorithms. So we start with 50,000 measurements. We discover the molecular basis of disease. And then we interpret this in each new biopsy. We interpret the rejectionness, including non-adherence. We're trying to project the clinical scenario for each patient. And then we also want to look at the wounding and atrophy fibrosis, because that's the principal determinant of outcome. Now we use fresh RNA or RNA, which is stabilized in RNA later, which is essentially fresh. So when we have two specimens from the same biopsy and we do them on two different chips, we get a line. If we have one fresh RNA and one formalin fixed RNA, we get many false positives and false negatives. You can use RNA isolated from paraffin, but it's covalently changed forever. One of the advantages of having a sample from the patient is that you're going to have that sample forever. And now you can use many new platforms, sequencing, et cetera, on that patient 5 years from now and apply new knowledge using the existing biopsies. So it's a permanent record and it's much more accurate. And that's why we do it that way. We have a variety of ongoing clinical trials, and including kidney, heart, lung, and liver, and calibrating cell-free DNA in kidney and in heart. And the heart cell-free DNA study is just beginning. And these are the centers that work with us, and we're very grateful to the outstanding people who actually help us to interpret the data right around the world. And I'd like to have India on board. I'd love to have an MMDX center in India to process your tissues. So here's what you do. You take a piece of human, a tiny piece, much smaller than is required for histology. You put it in RNA later. That stabilizes it now at room temperature for a very long time. You put it in FedEx, you send it to the MMDX center. They isolate the patient's RNA, put a label on it, hybridize it to the chip. And then after washing and staining, the label can then be read with lasers and turned into a spreadsheet. And our software turns that into a report. And for heart and lung, this takes 48 hours. It takes 2 nights. And for kidney and liver, it takes 1 night. So it's 24 hours for kidney and liver. I think we can eventually make it 24 hours for heart and lung as well. But these are very small bites. So MMDX heart, we started off with the problem of the low kappa values for histology. So instead, we actually used the kidney information and went into heart, basically trying to read hearts as though they were kidneys. And we used unsupervised principal component analysis and archetypal analysis. And this is the report that we currently generate. And I read a whole bunch of these yesterday evening while I was waiting for this meeting. So this is a new patient projected into the reference set. Our current reference set for hearts is 1,320 biopsies. And this shows that where that heart is. And it's sort of at we call this area minor, and I'll explain that. This is antibody-mediated rejection, T-cell mediated rejection. And then in the third dimension, you see acute injury. But this biopsy only has minor changes. Then we have a variety of other measurements, and I look at all of these and then sign it out. And the technician does just as good a job as I do, so this can be signed out by an experienced technician. It doesn't need an expert. So here's what we're seeing. In 1,320 biopsies, we have antibody-mediated rejection, a very clear signal. There's lots of antibody-mediated rejection. T-cell mediated rejection, there's about half as much as there is antibody. We have borderline cases of antibody and T-cell mediated rejection, which are here. We have a few of these acute injury biopsies, but we have lots of no rejection, and this is the majority of hearts, and they are the pristine ones, which are basically normal, And then there are those with minor changes. Now, we've been interested in these biopsies we call no rejection, but we see minor changes. And what are they? So we've made a new discovery, which we'll be presenting at the next generation of meetings. We're calling these no rejection, but if you look carefully, the ones that have DSA often have very mild antibody-mediated rejection-like changes, including NK cell changes and gamma interferon changes, but it's very mild. We don't know the significance of this, but it's not zero. So DSA-related changes are much more widespread than we had thought they were. And that's a new discovery. Now, when we look at graft losses, and the X's here mark the graft losses plotted on our map of all these biopsies. So we see a lot of graft losses in hearts that we say have no rejection. And we're trying to understand this. This is probably related to parenchymal injury. We see graft losses in T-cell mediated rejection. Strangely, so far, we're seeing very few losses in antibody-mediated rejection within 3 years, indicating that antibody-mediated rejection should not be considered an immediate disaster for the patient. This is a surprise. So when we look at actuarial survival, the blue line is antibody-mediated rejection. The red line is T-cell mediated rejection. Which line would you rather be on? And that's statistically significant. T-cell mediated rejection is very bad for the cardiac myocyte, and antibody-mediated rejection, being a microcirculation process, is relatively well tolerated in comparison. Now, one of the things that we're focusing on is parenchymal injury, because actually the patient came to you for the parenchyma. And so we're trying to understand how the parenchymal tissue, the cardiac myocyte, is behaving. And what we can find when we look at injury, this is a separate dimension of the tissue, is that there's a population of 20% of the hearts that we have are sent that have a late atrophy fibrosis phenotype. These are probably stiff hearts. They have lower left ventricular ejection fraction, and they're a serious problem in risk of failure. Only less than half of them have any rejection, however, but the rejection might have been what caused this atrophy fibrosis. So when we look at rejectionness in all the hearts, we get this set of actuarial curves: antibody-mediated rejection, T-cell mediated rejection. A few hearts with early acute injury do quite badly. And this is significant, but not very. If we look at injuriness and just measure the injuriness and reclassify all the same hearts, we have a highly significant difference where the severe injury and the late injury are the main drivers of outcome. And moderate and mild injury are relatively well tolerated. And if we just take all the hearts that have no rejection, so we take all these rejection hearts out, and now the hearts with no rejection, and then we look at the injury, and the injury is a huge driver of outcome in the hearts with no rejection as well. And it's the late injury which is the important thing there. And if we look at the histology classes, we can't see any significant prediction of survival. Let's look at lungs. We look at the transbronchial biopsies and the mucosa biopsies. This is from one patient. This patient has no T-cell mediated rejection. We generally find that the transbronchial and mucosa biopsies agree. In this case, we've got some injury, which we can see in the transbronchial biopsy. And in the mucosa biopsy, we're not seeing injury. Neither of them has any T-cell mediated rejection. So to summarize, because we don't have much time, in the TBBs, and we've got getting on towards 700 of these, we can see T-cell mediated rejection very clearly. Our new discovery is that there is a mild DSA-related change that looks a little bit like mild antibody-mediated rejection with interferon gamma release in the biopsies that are DSA positive. So we can't, although we can't define an antibody-mediated rejection phenotype per se, we can see antibody-mediated rejection-like changes, which may be stressing the tissue. And we can now diagnose clad-related changes. And these are in the alveoli, but they're more prominent in the airways. In the lung mucosal biopsies, we've been focusing on the mucosal biopsies because they're safer. These are from the third bronchial bifurcation, and they're safer. They can be done in sick patients. So we think that the future may well be, at least in part, in mucosal biopsies. In mucosal biopsies from the third bronchial bifurcation, we see T-cell mediated rejection. We actually see it better because this is more reproducible biopsy than the transbronchial biopsy. There's less piece-to-piece variation. We do see mild DSA-related changes, but there's not as much microcirculation in the mucosa, so we don't see this as well as we do in the TBBs. But we do see better clad association changes. And in addition, we also see not only atrophy fibrosis, which is the driver of clad tissue remodeling, we also see an inflammatory component that we don't see in the transbronchial biopsy. So let me just show you a little bit of that. In terms of searching for antibody-mediated rejection in lungs, the methods that we used that brought out kidney and heart right away do not bring out an antibody-mediated rejection phenotype in lungs, although there is a DSA-related molecular change. So we're trying to establish the significance of this mild change. And it's predominantly interferon gamma effects and inflammation. This is seldom suspected by the clinicians or pathologists. So in all the patients we've received tissue from, the clinician and the pathologist have very seldom, like it's only about 5%, have said that we suspect antibody-mediated rejection. So it's more common than is suspected that there is a mild change. And there's no strong association of these antibody-mediated rejection changes with clad. That doesn't rule out the fact that antibody-mediated rejection could be the stress which has led to clad in some cases. Of course, one of the problems with lung biopsies, particularly transbronchial biopsies, is that the heterogeneity may override some of these signals because the tissue heterogeneity is a big problem in all biopsy assessment. Now, in the analysis of clad, what we're seeing is clad-related molecular changes are best observed in the airway biopsies. It's a more prominent phenotype, and this is no surprise to those of us. I used to follow lung transplant patients back in the 1980s when we first were doing lung transplants in Toronto, and I remember seeing the first clad patients. So this is predominantly an airway disease, but it's also seen, as you know, there's another phenotype in the alveoli. And in the airways, we see injury and inflammation. So if we look at this is the advantage of having 6,000 biopsies in the can, all analyzed with microarrays. So we look at kidney and liver and lung and lung, mucosa, heart, and transbronchial biopsies. We see most of these, we're now measuring injury changes. And this is a log scale. So in the early period post-transplant, all the organs, probably not the TBB so much, but all the organs have an early injury signal, which regresses. It heals. And then something goes on in the lungs that doesn't go on in the other organs, both in the mucosa and in the transbronchial biopsy. So that's new injuriness occurring after several years in the lungs, which we're not seeing. The lungs are not stable like the other organs. This is liver, kidney, and heart, lung mucosa, lung transbronchial biopsy. And if we look at gamma interferon effects, again, liver actually has very prominent early gamma interferon effects. And late, the mucosa starts showing gamma interferon effects that we don't see at that level in the other organs. So the biological characterization of clad. What is clad at the molecular level? Well, in transbronchial biopsies, we see this. This is pathway analysis. Now, we won't go through this because I barely understand it myself and it would take all day. But we see what effectively this amounts to is tissue remodeling, atrophy fibrosis, stress response, but little inflammation. That's in the alveoli. In the mucosa, we see a different picture. We see many aspects of tissue remodeling and changes in metabolism, loss of metabolism. But we also see up here inflammation, gamma interferon effects. So we see an immune response, and this may be innate immunity rather than adaptive immunity. And some of these cases have rejection, and in many of them we can't actually say that they have rejection. We certainly see tissue remodeling and prominent epithelial changes because the mucosa is basically an epithelium. So to summarize, in heart endomyocardial biopsies, we can very clearly assign and quantify T-cell mediated rejection and antibody mediated rejection. We can also quantify acute injury and this atrophy fibrosis, probably stiff heart phenotype. The main determinants of left ventricular ejection fraction are injury and also T-cell mediated rejection, which impairs left ventricular ejection fraction. The main determinants of 3-year post-biopsy graft loss seem also to be injury and predominantly T-cell mediated rejection. In lung transplants, as assessed in the transbronchial biopsy, we see T-cell mediated rejection and DSA-related ABMR-like changes at a low level, and we see clad changes. In the mucosa biopsy, we see T-cell mediated rejection, again, very mild antibody-mediated rejection changes associated with DSA, but very strong clad changes. Prognosis in lungs seems to be due, if you've got T-cell mediated rejection, this is really bad. But most of the prognosis is determined by clad. And in multivariable analysis, clad wins. And so molecular clad becomes a very strong predictor of outcomes. So these are the brilliant people that have helped us in this project. And I'm here to talk about what they do, and they're the smart ones. And so I'd like to thank them, including my sons who work on the project, and they're both professors in our department. And this is a support. I'd like to thank the Mendez Foundation in particular for helping us with the lung studies. And I'd like to thank, above all, you for letting me talk to you. And we look forward to establishing MMDX in India. So thank you.
Thank you, Dr. Halloran. It was a wonderful talk, masterclass, and all of us have benefited, a lot of new ideas. I just request you to just stay till the end for the discussion, because I am sure there will be a lot of questions.
Yes, I will. Thank you.
Thank you. Dr. Palak Shah would be the next speaker on miRNA and heart transplant monitoring. He's a senior transplant and heart failure cardiologist at the Innova Heart Failure Group, Innova Fairfax Hospital in Virginia, US, director of the Innova Cardiovascular Genomics Center. And a lot of publications he has to his credit. I welcome Dr. Palak Shah.
Great. Can you guys see that okay?
Yep.
Okay.
Great. Okay. Thank you. I appreciate the opportunity to be invited to speak to you guys today about some of the work that we've been doing on circulating microRNAs, specifically Dr. Ravi Kumar and Dr. Patel and the other organizers of the meeting. I traveled to India over a year since I was probably about 5 or 6 years old. So it's really kind of exciting to hear about some of the things that you guys are doing and increasing access to organ transplantation for patients across India. So here's my disclosures. The ones that are relevant are grant support from the American Heart Association, my own institution, as well as the NIH for this microRNA work that we've been doing. So over the next 15 minutes or so, I will go over a couple of different learning objectives. One is to describe how circulating microRNAs can potentially be used as biomarkers for acute allograft rejection in solid organ transplantation. We'll talk a little bit about endomyocardial tissue biopsies and the microRNA signals that are seen during rejection and how those can be potentially used again to more accurately discriminate the subtypes of rejection when we evaluate endomyocardial tissue. And then finally, I'll talk a little bit about the work that we've been doing, which is focused on microRNA sequencing, again, with this goal of developing non-invasive biomarkers for not only acute rejection, but chronic rejection, as well as overall immune status of our heart transplant patients. I'm a heart transplant cardiologist. So since we haven't talked about microRNAs at all during this session so far, I'll just kind of briefly describe microRNA biology and how they work and why they're kind of intriguing biomarkers as well as potential therapeutic targets. So our body, our human body, has about 2,200 microRNAs, just like we have about 25,000 genes or so. They're encoded by our DNA, transcribed in the nucleus, and then exported to the cytoplasm, where they undergo a number of chemical modifications. Most importantly, though, they form this thing called the RISC, which is the RNA-induced silencing complex, where the microRNA binds to a number of proteins in the RISC, and then that RISC complex actually targets mRNA. And so it targets the 3' untranslated region of an mRNA. The RISC complex essentially will bind to that mRNA. And the net result of that is that that gene, that mature messenger RNA, is either destabilized and does not lead to protein production. So it essentially represses protein production. And when you look at it, about 50% of genes are under the control of microRNAs, and often multiple microRNAs can target the same gene. So this is a natural, biologically way that our body regulates gene expression. And the scientific community has kind of leveraged this and has developed actually novel therapeutics that are designed to mimic the naturally occurring RNA interference that microRNAs do. And so this concept is called small interfering RNAs. And there's 2 drugs that are that have completed development and are close to either approval or are already in clinical use. One is inclisiran, which is a small interfering RNA that targets the PCSK9 gene, and patisiran, which is a small interfering RNA that targets the amyloid precursor. And so the idea here is that they will target the gene once they bind to that complementary genetic region on the mRNA through Watson and Crick base pairing. It destabilizes that mRNA, and then the abnormal protein is not produced. In the setting of PCSK9, the PCSK9 is degraded. That leads to increased LDL receptor availability. Increased LDL receptor availability on the liver leads to lower serum LDL levels. So this is something that is currently being used and is also a way that we are targeting novel therapeutics, not only in cardiovascular disease, but in other disease states, including cancer. So the initial work, at least in cardiac transplantation, on microRNAs really comes from the Paris transplant group, and I know Dr. Lupi is going to be speaking to us soon. And what that group did is they did a case control analysis where they had paired tissue and serum analyses from 113 cardiac transplant recipients. They pre-selected 14 microRNAs based on publications prior to their work that were potentially mechanistically indicated or had been served has been shown as biomarkers in renal allograft rejection, and they evaluated them by PCR. And what you see here is some interesting kind of initial literature that suggests that certain microRNAs may be differentially regulated in the setting of either TCMR or AMR. As well as what I thought was very interesting is that certain ones seem to correlate more so with TCMR and other ones seem to correlate more so with AMR. They had performed tissue analyses as well. And one of the things you can see is that you can stain for some of these microRNAs specifically in endothelial cells, so that microRNA 10A or 92A seem to have potentially an endothelial cell origin that you're able to detect in the serum. They divided their cohort into a training and validation cohort, and you can see here that these microRNAs at least performed quite well at detecting rejection in their analysis. And this was really the kind of the impetus and the nidus for the work that we have been working on really since 2015, 2016. Aside from looking at circulating microRNAs, a group from Italy performed microRNA sequencing in endomyocardial tissue biopsies, kind of building on the work that Dr. Halloran just talked to us about to say, well, can we use these microRNA signatures to potentially enhance our phenotyping of subtypes of rejection when we're looking at tissue samples? They sequenced through next generation sequencing all 2,200 microRNAs, and then developed a kind of a list of 187 microRNAs that were candidate microRNAs that were then evaluated by PCR. And overall sample size is relatively small, but they had ACR, AMR, as well as mixed rejection. This is the unsupervised hierarchical clustering of the sequence data, and I'll show you some more data here. And what they basically did is did the comparisons to see how the microRNA signatures compared between ACR and mixed rejection, ACR and AMR. What they found is that mixed rejection and AMR seem to have an overlapping microRNA signature on endomyocardial biopsy. And I think this was kind of initial preliminary data that suggests that potentially this would be beneficial. Maybe it enhances the ability to kind of phenotype subtypes of rejection on endomyocardial biopsy tissues. And I think their technique allowed them to use formalin fixed tissue, so that didn't necessarily require kind of early processing and RNA later preservation, but you could do it after you had a tissue biopsy that already had shown rejection, and you're trying to better discriminate what subtype of rejection was present. So it's with this background that I've been honored to work with Dr. Hannah Valentine, who's pictured there, and a group of investigators from the Genomic Research Alliance for Transplantation. This is Dr. Valentine, who had been at Stanford and had done some of the initial work around cell-free DNA. When she was there, came to the NIH and developed this Mid-Atlantic Transplant Consortium for both heart and lung transplantation. And so it has 5 heart transplanters, of which Inova, where I work, is one of them, as well as some other regional transplant centers. And it was a multi-center prospective a clinical study where patients before heart and lung transplant were enrolled, and we had serial blood samples collected before transplant and after transplant for up to 5 years. There we go. So for our circulating microRNA work, we are doing next generation sequencing of microRNAs. We use the GRAPH cohort, and the data that I'm going to present to you is a case control study format, but we are working on sequencing a larger number of samples, so that right now our data set is about 200 to 300 samples, but we're working to sequence, hopefully by the end of the year, about 800 to 900 samples. We were lucky enough to work with Dr. Kieran Cush at Stanford for a validation cohort as well. So overall, it's 109 heart transplant patients, of which 68 are from Graft and 41 are from Stanford. 65 of these patients are controls. These are patients who have had no history of allograft rejection after transplant for at least a minimum of 1 year follow-up. And then there's a subgroup of patients with both TCMR as well as AMR. When you look at the subtypes of TCMR and AMR, the majority of patients have kind of moderate rejection. The incidence of severe rejection, either INSHLT grade 3R cellular rejection, as well as PMAR3, really we didn't see much of. Here are the patient characteristics overall. Generally speaking, I think most of these kind of patient characteristics are relatively similar. I think the big things are that younger patients seem to have a much higher incidence of rejection than older patients. The other thing that we saw, which I thought was very interesting, which may be a little bit less relevant in India, but certainly what's been seen in America, at least, is that Blacks or patients of African ancestry seem to have a much higher incidence of antibody-mediated rejection. And we can talk about that a little bit more during the discussion. And then patients who are bridged to transplantation with the left ventricular assist device also seem to have a higher risk of sensitization and antibody-mediated rejection. So in terms of our sequencing, we sequenced, again, plasma samples here, generated about 11 million small RNA reads, of which about 50% of those are microRNA reads. About 450 microRNAs are expressed in the plasma. This is pretty consistent. So even though your body has about 2,200, you only express about 500 or so in any given tissue. And then when you look at the abundance of microRNAs, really the top 20 microRNAs, which are depicted there, account for almost 75% of the expression that we see in the circulation. So we performed a differential kind of microRNA analysis for TCMR and then AMR. In the setting of TCMR, we saw certain microRNAs that were upregulated at the time of rejection, but didn't really see any that were significantly downregulated. On the other hand, with antibody-mediated rejection, really it's a lot more kind of diverse in the sense that you have certain microRNAs that seem to be upregulated, other microRNAs that seem to be downregulated. And there were a number of microRNAs that overlapped both TCMR and AMR that weren't present or differentially expressed in controls. So one of the things that we've seen quite a bit is that TCMR seems to be something that has a little bit shorter onset, comes on quickly, we treat it, and then often the signature for that resolves. And when we looked at the microRNAs, what you can see is that if you look at the differentially expressed microRNAs prior to T-cell mediated rejection, you really don't see a significant alteration in gene and microRNA expression. At the time of rejection, you can see a number of the microRNAs are upregulated at the time of the event. And then when they're treated for rejection, the microRNA signature seems to normalize, but not completely. And this is very similar to if you look at cell-free DNA, which is depicted on the bottom, where the cell-free DNA measures right before an episode of rejection really seem to be otherwise relatively normal. But then at the time of rejection, you can see that the cell-free DNA levels increase with at least cellular rejection. On the other hand, when you look at antibody-mediated rejection, it's a very different pattern, where before antibody-mediated rejection, the microRNAs of interest really seem to be kind of aberrant and dysregulated compared to controls. They remain aberrant and dysregulated in the setting of the actual rejection, the endomyocardial biopsy proven rejection. And then after we treat them with things like plasmapheresis and IVIG and B cell agents, you can see that there is some normalization of that microRNA signature, but certain microRNAs seem persistently dysregulated after treatment for AMR. And this is similar to what we see with cell-free DNA, where the signal and the injury signal for AMR seems to precede the diagnosis of AMR, often by months. This is another way of looking at this. This is a single individual patient with multiple microRNAs that were mapped serially after transplant. It's almost like what we do with DSAs. And you can see that certain microRNAs are upregulated at the time of antibody-mediated rejection, others at the time of T-cell mediated rejection. And this really shows that one microRNA is not going to be the right signature. You're really going to need a panel of microRNAs to be able to differentiate the subtypes of rejection. We performed a pathway analysis, and like Dr. Halloran, I don't understand all of this, but this is a way of identifying what the messenger RNA targets are of the microRNAs that were expressed, differentially expressed. And we can see that the gene targets for TCMR and AMR are actually relatively distinct, but there is a group of about 35 genes or so that seem to overlap. And the majority of the overlapping genes are really involved in interleukin signaling, TGF-beta signaling, cellular apoptosis, cytokine, really inflammatory signal. So we validated this work in the Stanford cohort, and what you can see here is that the performance of these microRNA panels for acute rejection, TCMR and AMR were quite good in this relatively small sample size patient population that we had, with an area under the curve that approached excellent at 0.88 for AMR specifically, and was slightly lower for TCMR. So there are some limitations of this work. We realize it's a small cohort size, and we only, again, sequence about 300-400 patients, so we're working on much larger cohort size and a distinct validation group. The prevalence of rejection is low, and I think I know that this varies from center to center and country to country, but overall in our cohort at Graft, we're seeing a prevalence of rejection of about 5 to 8%, so relatively low prevalence of rejection, which makes these types of analyses difficult. We had rejection that started at 8 days all the way out to 2.5 years. So we were unable to describe performance at different time points just because, again, of a smaller sample size. We're unable to differentiate moderate versus high degree rejection. I think that's where a biopsy or molecular microscope or some other injury marker will be required. And at the end of the day, we perform these analyses, and we're really using this imperfect gold standard, and really need to do this work with the molecular microscope or cell-free DNA or some other injury marker. So in conclusion, I think we feel confident that there is a distinct circulating microRNA signature in plasma that can differentiate AMR and ACR, or T-cell mediated rejection. These microRNAs likely reflect some degree of the graft injury that's actually occurring, but also the systemic response or the immune response to injury, to rejection. And like we talked about a little bit more, that the endomyocardial biopsy really is an imperfect gold standard. And we're working on trying to validate these microRNAs with other more sensitive markers of allograft injury or of the individual subtypes of rejection. And, you know, I think one of the things that's important for us to realize is that, you know, a lot of our energy in our discussion today is on rejection. But I think a lot of the problems that our patients are dealing with, and this is what Dr. Karen was talking a little bit about, is that we need to start thinking beyond just the detection of rejection and the prevention of rejection, which the overall incidence with modern day immunosuppression is relatively low, and start thinking about how we can use these molecular technologies to prevent the long-term consequences of transplantation and immunosuppression. So I just want to thank the people that have supported me both with funding as well as mentorship, resources and scientific collaboration. Dr. Hannah Valentine, Sean Agborino, who's an intramural investigator at NHLBI, who's focused in lung transplantation, and Dr. Karen Cush, who's over at Stanford, as well as others who aren't depicted here. Look forward to talking to you guys all at the discussion, and appreciate the opportunity to present.
Thank you, Dr. Palak. Wonderful presentation. A lot of new knowledge seemed to have come and probably would be interesting to discuss. And now the next speaker is Professor Alexander Lupi. He's again one of the giants in the field of molecular science in heart and transplants. The group heads the Paris transplant group, and also is a professor of nephrology at Necker Hospital. Is an expert for multiple organizations all over the world in this field, and principal investigator for novel research projects like iBox, eye transplant, etc. So he's a very well-known name. He's also, I would say, for the Indian audience, he's one of the who have been awarded the Paul Thoracic Award from the American Society of Histocompatibility, along with Dr. Halloran. So, Dr. Lupi, please be welcome.
Thank you very much, and thank you for the very kind invitation by you, Dr. Ravish Kumar, and Dr. Jig Patel and the INSHLT. My talk will be covering some aspects of heart transplant diagnostics and how we can push some evidence coming from science and transform it through innovation. And I will be covering some of the next steps on the behalf of the Heart Transplant Diagnostic Innovation Consortium. And I would like to start with just thanking all these people, because it's a very wide consortium composed by heart transplant surgeons, cardiologists, very dedicated heart transplant pathologist experts, and data science people, and nephrologists like me. Okay. So what do we want to achieve? We want to achieve precision pathology, which means measuring objectively relevant pathology components in tissue. And we want to reveal mechanism, best choice for therapy, response to treatment, and try to think about having surrogate markers for outcome. And I would like to start this talk by, just stepping back for a moment and understand the lessons from the kidney and how we can learn from kidney for heart application. And it's going to be about how we can envision integrative approaches, but also how new technologies, analytical platform can help us, and how these are now occurring for concrete achievements. And this is the global scheme in the Paris Transplant Group that we have been building over the past 50 years now around kidney transplantation, just because we are collecting prospectively almost every possible data from patients, starting from biomarker, histology, environmental factor, any therapeutic change. And this allows us to have this map of patients that we can integrate into our pipelines and databases system. And only having that deep level phenotype cohort allows us to build relevant prognostic modeling and diagnostic modeling. So this is where the biggest effort should be made if we want to transform knowledge into products that clinicians will be using for the benefit of patient care. And I really would like to make that point very clear here, that this kind of initiative is a very hard work on a daily basis to be sure that all our prospectively collected data are actually meet the standards for that kind of data collection. And we were asked by the New England Journal, to summarize our approach that we have been doing in the past now 10 years, and how we could see the differences, but also maybe the overlapping pathways between organs and how one organ could inform another. And you have had some examples in the previous talks. But really, when you think about kidney, heart, lung, and they did not allow us to put liver already, because I think that the knowledge has to be improved. But we are starting to better understand how the organs behave into the alloimmune injury, and also our heart transplant patients can have different patterns than what we observe also in kidney. So we have differences, we have similitude, we have discrepancies, but I think overall we can learn a lot from each other. And this is also from the New England Journal paper, and it's a slide summarizing also a very important concept is how we can transform heterogeneity from a weakness to a strength. We are doing research in humans, not to say that research in mice is not useful, but we are not treating mice at the end of the day. So I think that nowadays in the 21st century, we can acknowledge the power of human studies. When you have this deep level phenotype cohort and you have the information about tissue injury, about what is happening in the blood and what the patient outcome is in a very standardized manner, we can draw very meaningful conclusions, not only for diagnostic purpose, but also for pathophysiological enhancements. So it's a kind of different setup of the global scheme of research. We are not starting from basic science and we go to humans. We are working closely with humans, and maybe we generate new hypotheses for the basic science field. And coming back into the biggest problem is how we can move in heart transplant pathology into a more precision diagnostics based approach. I think we have to achieve and take into account the very integrative approach that we need to do, starting from, of course, light microscopy, HLA, but also the biomarkers that have been presented before, chemistry, clinical history, molecular diagnostics, and immunofluorescence to achieve a final diagnosis. So you see that our task is quite big. And I just wanted to take 1 minute in just that slide because I think it's important. One of the biggest mistakes in the transplant field, to my point of view, has been the disconnection between the non-invasive biomarker and the allograft precision phenotyping. So you had people who say that biopsy was not useful anymore because we had these fancy non-invasive biomarkers. You had also people from the pathological side who said, non-invasive biomarkers are not useful for precision diagnostics. It's just for allograft monitoring. So I think that we have to have a different vision. We need to have a reconciliation of those two approaches because I think they are very complementary in the way we are actually managing patients in the daily routine care. We know that we are doing too many unnecessarily biopsies. And this is where non-invasive biomarkers like, microRNA, donor-derived cell-free DNA can be useful. But once a biomarker shows up, we have to perform a biopsy. In this case, we need complementary and companion tools with molecular technologies. So instead of separating these two approaches, we try, to reconcile them, and we try to put them together in nicely annotated studies. And that's actually, I think, where genomic medicine has to move. So, we published this paper a few years ago in Circulation with Phil, and it was a very nice, story because at the beginning, there was no signal or anything in heart transplantation running the gene expression profiles. And we try to have a different approach. So what we try to do is we use our heart transplant pathologists to reassign diagnosis. This is where some molecular signals, showed up. And then we had this spectrum of injuries in heart transplantation, and we published that in Circulation. And this is the editorial from John Kobashigawa, The Search for a Gold Standard to Detect Rejection. Are we there yet? I think the answer is no, but actually I tend to see the glass half full. This was a multicentric study in 5 French referral centers, and we screened patients for heart ABMR, and we had matched controls. And again, I think all these data just came because we had these deep level phenotype cohorts. And we had a very careful histological phenotyping platform, which is summarized here. And we also had for every single patient anti-HLA DSA characterization. And we added on top of that the molecular phenotype and using the Edmonton approach with transcript PBTs and classifiers. That allowed us to have this landscaping of the relevant and mostly significantly differentially expressed gene between AMR versus everything else. And you have here the volcano plot with the 9,000 IQR filter genes and the hits that you will recognize: CXCL4, CXCL11, CXCL10, PLA1A, and a different of these genes. And actually, if you take the top 30 canonical pathways annotated with the ABMR selective transcripts aligned by p-value, you will recognize gamma interferon genes, NK genes, macrophages genes, and also endothelial genes. So we observed the fact that allograft gene expression correlates pretty well with AMR diagnostics, not perfectly, which I think is good. And also, more importantly, we saw an increment with the increase of the ASHLT AMR grading, starting from PMR0 to PMR1I+, H+, and PAMR2 and 3. This increment on NK burden, EDSAST, interferon gamma inducible transcripts. So I think it was a very important study on a carefully annotated data set, supporting the fact that we had some kind of molecular architecture in heart antibody-mediated rejection. So is it, a standalone test? I think no, we need to go further. And, from innovative research to scientific and clinical validity, we are talking here about the life cycle of diagnostic tests. We need more than that. We need proof of clinical utility, technical optimization, and then last not least, the FDA approval or lab developed test. And on top of that, we need approval for payment by payers, and we need technical optimization. So I think there is a long road ahead, but actually we have already, I think, some relevant data. And we also need consensus. You hear many things about many technologies, many indications, many applications in tissue, in biopsy, applications in body fluids, and everyone will be talking about different methods, targets, platforms, centralized, decentralized, and on top of that, you have the complex bioinformatics and how we deal with that mass amount of data and phenotypes. So we need consensus on that, and I think we do not have any consensus. We know that we have many transcriptomic techniques. The microarray started 30 years ago, and we have next generation sequencing. We are ourselves doing a lot of NGS, and those are techniques which need amplification, and you have also the nanostring technology that I will be talking a little bit more in detail in the upcoming slides. So if you compare, the FFP-based technologies with fresh tissue of DNA microarray-based, you can achieve a fair amount of transcripts using the nanostring technology. We have, the company allows like 800 transcripts to be tested. You have this table comparing the two technologies of the shell panels available. You can do custom panels with encounter of even microarray. This is the recommended RNA input quantity. Interestingly, the FFP nanostring technologies doesn't need any reverse transcription and amplification, which is, to my point, a big advantage of that technology. The turnaround time is pretty much similar. The manufacturer allows a software, but it's very complicated for microarray. It's pretty easy with the encounter technology. And two important points. What is the ability of the technology to use sample for histology and gene expression analysis? Of course, one of the biggest advantage in FFPE is that you know what you are looking for. And I think it's a very important advantage. And also, you can have access to the long-term clinical follow-up data on archival clinical samples, which means that you can build your reference set in a very, very small amount of time. So also the nanostring technologies platform is approved by FDA, and the price is, you can see here, pretty low. And also it allows us to move from a centralized to a decentralized workflow, which I think it's a big technological disruption in the way we are now envisioning the future. So what's the next step forward? The next step forward is to be hot, which means the BANFF Human Organ Transplant Panel. It's about 770 genes, and I will be describing this multi-transplant organ panel. So first of all, the BANFF classification started to think about this multi-organ gene panel, and now the effort involves many different platforms from different countries, which are summarized here. And we did a lot of work doing systematic reviews, text mining, and the BANFF consortium also worked very hard to end up with this BANFF Human Organ Transplant Panel, and you can have details in the publication in HAT in 2020. And this is what you have in this panel, and as you can see, it covers 770 genes related to hematopoiesis virus. On top of that, we added the SARS-CoV-2 recently. You have also different like BK and CMV, apoptosis, inflammasome, cell cycle, and also organ-specific genes for lung, kidney, heart, and liver. And very importantly, all the genes related to the immune response, and all the BHOT panel is summarized here. A very important step has been the discussion not only in the kidney field, but among the merge of BANFF and INSHLT. You will recognize very key opinion leader here, like Dylan Miller, Gerald Berry, Jean-Paul Jung, of course, Dr. Fishbein, Ornella Leon, Delse Neal, and all these people. And they published this report on the allograft pathology, and this BANFF workshop on heart report, I think, is amazing and paves the way for the future. And also, when we think about the next generation, not only technologies, but clinical trials, we have to think, to my point of view, differently. We have to move from a very centralized and one center doing everything to the fact that we can have very collaborative initiatives, which means that we can build platforms for data repository. And to my point of view, this will enhance collaboration, foster discovery, and this is the way we will end up with a very relevant diagnostic system. We will all work together. We will all build an open source, open access platform with pipelines and analytical schemes for all these efforts from different platforms to be put together for the same initiative. So that's the ICDOT initiative. It's International Consortium for Diagnostics and Outcome in Transplantation. We started with kidney, but we are moving also now with heart transplantation, lung and liver. And you can see here that we have the pathological department from University of Alberta in Canada, Imperial College in London, the Mass General Hospital, and our own institutions. Participating groups, importantly, will have secure access to anonymized data to facilitate the joint effort to validate the tissue-based molecular nanostring testing for transplant care. So I think this is very important to see that there are options for changing the global schemes. And this is the ICDOT global initiative, from data production to data integration and how the results will be handled. So now I'm talking about the studies that we have started 2 years ago. And just to show you that we will keep the same degree of phenotyping with all our pathologies, reviewing independently many slides which are digitalized. And on top of that, we are paying particular attention to build heart transplant very precise and deep phenotype databases. We have involved so far 932 patients, and it's going now full speed. And I hope I will be able to provide you some results pretty soon of this initiative. So about the conclusion and next steps, I think collaboration is needed, of course, but also we need to kind of change the scheme. We need a centralized data collection platform. The BHOT panel has many possibilities, but will be developed to facilitate standardized research collaboration. And we are thinking about a complete decentralized system, which this technology actually allows. And last but not least, we are working really hard and we have big grants to be able to home and build this open source multi-center database to validate molecular diagnostics and to the end of moving towards therapeutic decisions and clinical trials. This is how this community is trying to build this platform with refinement of classification and how new members like biostatisticians and omics specialists and biomarkers will be enrolled, as well as the need for AI and data science and the way we will handle this very complex mass amount of data. One example we published this year, actually last year in 2020 in Circulation, was the prototype of integration and AI-based modeling in heart transplantation. And you can see here the paper. It's about cardiac allograft vasculopathy and how we can predict trajectories of heart transplantation. And this is the editorial written from Dr. Moayedi and Tedeberg. And the Paris Group Molecular Platform is available to the network for transplant diagnostics. We have 12 full-time equivalent people just dedicated to the platform. And importantly, we have the control on the pathological core because it all starts with pathologists who are versed into the heart transplant field to be able to read biopsy and apply the criteria and understand the relationship between the standard pathology and the molecular signal we can have. And I'm grateful to Jean-Paul, Patrick Bulval, and Alaise Angelini in Padova. We have an operational management, engineers, PhD students, bioinformatics, and also interfacing and cloud computing and some research assistant too. And this is now how the Paris Group Molecular Platform is operating and is available to all of network. We have a lot of grants going on, so we are very keen for a lot of collaborations. And I hope that this society and our friends and colleagues in India would like to make this collaboration with us, as well as other centers in Europe now, United States and Canada. So I would like to thank you for your attention. Again, thank you for the invitation. I'm also very grateful for the collaboration we have with Cedar Sinai and Dr. Patel, who is connected. I think it's very early for him. And in case you want anything, and if you have questions about the platform, about what we are developing now in heart, and even some of our preliminary results, don't hesitate to contact me. Thank you very much.
Thank you, Professor Lupi. A very wonderful presentation, and we look forward to further development in nanoheart design or the IC.dot platforms. We will be tracking you on Twitter, as you said. Hopefully, I think we look forward to a lot of exciting developments from your end. So very thank you for joining us. I hear you are very busy, maybe for some other engagement. So if you possible, you can join the discussion.
Thank you very much.
Our next speaker is Dr. Jignesh Patel, co-medical director of the heart transplant program, Cedar Sinai, and one of the senior-most world experts on field of immunosuppression and heart transplant. He's also a long-standing faculty of the INSHLT, was ex-chairman of the heart transplant committee. So over to you, Dr. Jignesh.
Thank you so much. Let me see if I can get my slides up here. Can you see my slides okay?
In the small format, probably full screen.
Are you seeing it on the full screen now?
Not so, not one. Yeah, so,
There you go.
Yeah.
That's on the full screen now?
Yeah.
Great. Let me see. So this morning I'm going to speak a little bit more about cell-free DNA in both heart and lung transplantation. We've already heard from Dr. Kim yesterday regarding a single center experience with this technology, and also this morning from Dr. Kaur regarding the technology itself. Well, the endomyocardial biopsy has really been the gold standard for rejection surveillance in heart transplantation for several decades now. And the main issue with it is that you have to perform the procedure frequently, and it's an invasive technique, not free of complications and patient discomfort. But there are other intrinsic problems of the current pathology approach to the diagnosis of rejection. The definition of lesions is based on expert consensus and not clinical outcomes. The grading is modeled on morphological features and not on functional or pathological evaluations. For example, if we see lymphocytes on the biopsy, we do not know what they're doing, if they're exerting cytotoxic activity or just present. And traditional histological methods are often subjective, with concordance among pathologists as low as 28% in the Cargo study for 2R rejection, due to equivocal findings with things like old biopsy site, quilty lesions, and infections, which I'll show here. And you can see on the slide on the left here that an old biopsy site, bioptomes generally tend to go to the same place when you take a biopsy, and you may see myocytes in disarray, and if the biopsy was recent, you may even see some inflammatory cells present. Infection can certainly complicate the issue, particularly with CMV and toxoplasma showing lymphocytic infiltrates. We actually just last week had a patient with toxoplasma in the heart, but the biopsy was initially read as 2R, so that can be quite confusing. And then there's something called the quilty effect, which was first described by Dr. Margaret Billingham in 1981, which occurs in up to 20% of biopsies and is characterized by a lymphocytic lesion with B and T infiltrates, but really is not a rejection phenotype. And it is considered by some to be a side effect of immunosuppression, particularly cyclosporin. So there have certainly been innovations in rejection surveillance after heart transplantation and solid organ transplantation in general, as you heard from Dr. Lupi. And we heard earlier that defining rejection with molecular approaches appears to be a very promising approach, with intergraft mRNA transcripts from Dr. Halloran and microRNA from Dr. Shah. And we heard yesterday from Dr. Coutances regarding the cardiac iBox technology to predict CAV trajectories. I'm going to focus mainly here on acute rejection surveillance using donor-derived cell-free DNA. So it's important to know that cell-free DNA is found in circulating blood, and there is a basal rate of cellular turnover that results in a level of around 6 nanograms per mil of plasma cell-free DNA. But there are higher levels associated with any form of increased cell damage, for example, trauma. And cell-free DNA, when it with respect to solid organ transplantation, really leverages the observation that the allograft DNA is different from the recipient DNA, and this difference can be assessed by genotyping single nucleotide polymorphisms. And up to 266 SNP loci may be used to differentiate between donor-derived cell-free DNA and the recipient's cell-free DNA as well. And the fraction is what we are looking at when we're assessing rejection. And the initial study was really part of the Cargo 2 observational study, which looked at a rejection cohort and quiescent cohort. There was blood that was banked at the time of biopsies that were taken, and where there was concordance amongst pathologists for rejection versus quiescence, the blood was analyzed for cell-free DNA. And they found 28 patients in whom there was blood available prior to a diagnosis of rejection. And also there was a treatment effect cohort of 17 patients where blood was available following the treatment of rejection. And this was compared with 26 patients who were quiescent. And the original finding was that certainly rejection was associated with increased donor-derived cell-free DNA fractions compared to quiescence, with a mean cell-free DNA level 1.9-fold higher than the quiescent group. But you'll notice that there is a significant variation in terms of absolute levels. The other important finding of this early study was that cell-free DNA levels actually start increasing even before the pathological diagnosis of rejection is made. And these values tend to increase up to a month prior to transplantation. And importantly, treatment of rejection also leads to a subsequent decline in cell-free DNA levels. So this was a very important early demonstration that you can look at cell-free DNA for purposes of rejection monitoring. It can go up prior to histological rejection and may be useful for monitoring treatment effects. And we followed this up with the prospective AlloSure study in heart transplantation, which was an analysis of the DOR registry. This data was presented earlier by Dr. Kaur, so I won't belabor the point. But in this registry, there were over 400 patients with paired biopsy and samples, 18 rejection episodes with antibody-mediated rejection, 12 by surveillance, 6 for cause, 17 cellular rejection episodes grade greater than 2R, 2R or greater, 9 surveillance, and 8 for cause. And they were compared with a cohort of no rejection of almost 800 samples from 400 patients. And the findings were that certainly patients with cellular or antibody-mediated rejection had elevated donor-derived cell-free DNA fractions compared to patients with no rejection. And the quiescence rate, patients had a cell-free DNA level of around 0.06% compared to 0.17 for patients with rejection. And when they looked at severity of rejection, certainly the cell-free DNA levels increased with the severity of cellular rejection, although this was less, this was really not validated in patients with antibody-mediated rejection. And the study found that at a 0.2% threshold, donor-derived cell-free DNA had a 44% sensitivity to detect rejection, but importantly, a 97% negative predictive value. Interestingly enough, we did have a Cedars-Sinai cohort in this study, and our transplant cohort is notable for a very large proportion of patients who are highly sensitized. And in our specific cohort, what we found was that the basal level of cell-free DNA was actually significantly higher at 0.16. And those patients that developed antibody-mediated rejection had a subsequent rise in their cell-free DNA fractions to 0.5%. And we also did a study where we analyzed patients who'd had both donor-derived cell-free DNA samples taken and a biopsy where we did intragraft mRNA transcripts with the molecular microscope. We identified 16 biopsy samples from 10 patients with concomitant assessment. And what we found was that an elevated cell-free DNA ratio of greater than 0.5% in the presence of cellular antibody-mediated rejection was correlated with results of the intragraft mRNA transcript and pathology biopsy. And when we looked at this data, what we saw was that the donor-derived cell-free DNA blood samples correlated with pathology samples in only about 75% of cases, 7 no rejection and 5 rejection, compared to mRNA transcripts in 100% of samples using the molecular microscope, with 9 rejection and 7 9 no rejection and 7 rejection. In 7 cell-free DNA samples that were greater than 0.5%, 5 out of 7 correlated with pathology rejection, all AMR1, and 100% correlated with abnormal mRNA transcripts indicating either AMR in 6 cases or cellular rejection in 1. So the correlation between donor-derived cell-free DNA and mRNA transcripts appears to be much more robust than histology itself. I just want to really emphasize this very recent publication from Dr. Shah's group that came out in Circulation only last week, which is a very important study looking at cell-free DNA from the GRAFT consortium, where they had 200 patients that were analyzed, 171 transplant patients that underwent serial plasma samples and concomitant histology as well, and had assessment for donor-derived cell-free DNA in correlation with allograft rejection. And what they found was that certainly cell-free DNA was highest in the first couple of months post-transplant, but then the level really declined significantly and stayed down for up to 18 months before there being a slight rise again in the later population. And certainly cell-free DNA was associated with increased grades of cellular rejection and also antibody-mediated rejection. But more importantly, cell-free DNA also correlated with clinical outcomes, particularly with severity of left ventricular ejection fraction declines as well. And the greater the decline in ejection fraction, the greater the increase in cell-free DNA. And the group also found that the rise in cell-free DNA predated histological findings as well. And this was far more prominent for the antibody-mediated rejection cohort than the cellular rejection cohort as well. And this may certainly explain why basal levels of cell-free DNA are increased in AMR patients, which may also correlate with the increased molecular patterns that have been observed by Dr. Lupi. There were some other interesting findings, specifically with regard to antibody-mediated rejection. The group found that AMR patients had a much greater rate of small fragment cell-free DNA samples with fragment lengths less than 100 base pairs. And these fragments also tended to have a higher guanine cytosine content as well. So perhaps cell-free DNA may be able to not just tell us whether rejection is about to happen and is happening, but we may be able to even decipher type of rejection through use of these non-invasive samples. And you can see here that really that they also noted that cell-free DNA levels decrease with treatment of rejection, both cellular rejection and antibody-mediated rejection as well. And their AUC for acute rejection was 92%, which is really really outstanding, with a negative predictive value of 99.2% with a threshold of a cell-free DNA value of 0.25. So certainly this data further consolidates the prior experience in heart transplantation. The other findings that were of note was that 1 in 5 patients had allograft injury as defined by an elevated cell-free DNA. And the majority of patients who had allograft injury but had a negative biopsy were of clinical relevance. A fifth of them were associated with concurrent allograft dysfunction, and about half of them had preceding acute rejection or allograft dysfunction as well. So again, we see this pattern of the biopsy really not providing the full picture in terms of what's going on at a molecular level. I wanted to now move on to some data in lung transplantation. This study was recently published in lung transplant involving 190 lung transplants with about 100 patients analyzed for assessment of graft dysfunction and rejection. And what the group found was that certainly changes in FEV1 correlated with increased levels of cell-free DNA. Cell-free DNA levels were higher in patients who had donor-specific antibodies and also positive histopathology on biopsy. The authors also found that the changes in FEV1 particularly correlated with cellular rejection, and it was with antibody-mediated rejection, and it was the antibody-mediated rejection samples that were particularly correlating with elevated donor-derived cell-free levels. And when they looked at clinical outcomes with respect to allograft failure-free survival and CLAD, patients with the highest tertile of cell-free DNA had the lowest survival and the highest rate of CLAD following transplantation. There's another study that actually looked at cell-free DNA in bronchoalveolar lavage samples and also saw correlation with elevated cell-free DNA levels in those specimens with a greater likelihood of developing graft failure as well. So it looks like you can use this technology potentially for both blood and BAL samples. This is an interesting study looking at the utility of cell-free DNA in human ex vivo lung perfusate as a potential biomarker to predict the risk of primary graft dysfunction in lung transplantation. And while it's a small study, there does appear to be a signal for elevated cell-free DNA levels in samples in ex vivo perfusion platforms, although there seems to be a significant overlap. But this is certainly a promising application of this technology. So in summary, the biopsy has endured as an imperfect gold standard for diagnosis of acute rejection in thoracic transplantation. Non-invasive techniques or the liquid biopsy has been an attractive goal to facilitate easier monitoring for rejection. Donor-derived cell-free DNA provides a useful tool. It has a high negative predictive value for allograft rejection. AMR appears to be associated with higher levels than ACR, shorter fragments, and higher GC-rich fragments as well. The rise in cell-free DNA appears to predate clinical and histological diagnosis and may therefore allow preemptive treatment. Well, what are the future directions? The incorporation of cell-free DNA with a gene expression profile is certainly has reached the clinical stage, and we're participating in the SHORE registry to look at this data in greater detail. This really allows us to look at both the AlloMap and the cell-free DNA values for patients and make an assessment of what's going on at a molecular level. We're hoping to have more data on the incorporation of cell-free DNA with the molecular microscope, and hopefully with more samples, we'll be able to determine a correlation there. There may be really a future role of this technology for xenotransplantation, and certainly there are moves towards xenotransplantation reaching the clinical arena in the next year or two. The assessment of graft function in ex vivo perfusion systems, not just lungs, but also hearts may be useful there. And also, there is some data to suggest that chronic graft dysfunction may correlate with cell-free DNA levels as well. So a lot of promise here with this technology. The advantages have been outlined before, that this is something that can be applied relatively easy easily at local centers. So I wanted to thank you for your attention and really acknowledge our group at Cedars-Sinai for providing much of the data for the work that's been done in this area. Thanks again.
Thank you, Dr. Jignesh. Very nice presentation. You have summarized overall the evolution and clinical significance of this test, particular test. So if you just can wait for discussion. Before that, Dr. Howard Eisen had suggested that someone talks from INSHLT on the Indian scenario, just a very short brief on what is going on registry. So I would like to invite Dr. Professor Sandeep Seth, cardiologist, senior cardiologist from All India Institute of Medical Sciences, for his short presentation. Sandeep? Yes. Can't hear you. Can you unmute, please?
Okay, can you hear me now?
Yeah.
Okay,
Yes.
sorry, I didn't unmute myself. Yeah, so as I was saying that for about the last 2 years, Ravi, myself, Dr. Gokhale, and the other members of INSHLT have been trying to collect data on heart transplant, lung transplant, and LVADs in India. And this is part of the consequence of what we presented last year in August. So this data was updated till August last year. And the idea was that we would try and update this data at least once every 6 months to 1 year. And this is the presentation we presented last year. Some of this data is, of course, things with which you're familiar. Heart transplant started in 1967. You're familiar with this photograph. Along with that, the first transplant in India took place in 1968. Dr. P.K. Sen did a transplant. The patient did not survive, so very few people got to know about this transplant. The transplants all over the world took some time to take off because the initial transplants were not very successful till we got to have a number of good drugs, biopsies, and a whole lot of new techniques. In India, the first successful transplant took place in 1994 with Dr. Venugopal at AIIMS, and that was a success story in India. This was reported in 1994. Dr. K.M. Cherian did the first heart, lung, and the lung transplant. So that is history of heart and heart-lung transplant, lung transplant in India. Now, what about data? We have limited data on the transplant programs in India. It's only recently that we have started publishing data on the transplant programs. There is some data from the Apollo group. So we have some data on the heart and heart-lung transplants. I will not go into the details. We have some data from Dr. Balakrishnan's group on the growth of the transplant program. The initial transplant program has been very slow till about 2012. There were very few transplants being done. From about 2012 to 14. Suddenly the transplant programs started picking up, and now the transplant programs are becoming more and more prolific. The results of the transplant programs are also very good. This is again from Dr. Bala's group. The survival rates are also very good. This is from Dr. Ravi's group. This is our own data. This is the data when we started increasing our transplant program. The first 2 years, we found that as the rates of transplants increase, our complication rates increased. And then with the rates of infection, the rates of rejection going up, we changed all our transplant protocols. With that, in the next 2 years, all our complication rates started going down. So shows that if you are vigorous about looking at your data, keeping an idea about what's happening with your program, that vigorous look at your data improves your results. This is the same 2 years. The initial 2 years, when we suddenly started increasing our transplants, there was an increased rate in the complications. Then, as we changed our program, the survival rates improved. So what have we done in the past 2 years? The first step we did was that we encouraged all the centers in India to start listing themselves in a simple Google sheet. We just told all the centers, just list yourself in an open Google sheet, because till that point we actually had no organized system of collecting data in a centralized registry in India. So we actually had no idea how many transplants are being done in India every year, what are the results, how many heart transplants, how many lung transplants, how many assist devices are being put. So we actually had no idea. So what we could find out, there are at least 71 centers in India who are actively involved in doing transplants. So this is just a list of those centers. So there are at least 71 centers all over India who are doing transplants. So when you look at the numbers, these are the numbers. There are more than 1,000 transplants which have been done for heart transplant, more than 94 heart and lung transplants, more than 280 lung transplants. The numbers have increased since, and almost 100 LVADs have been put. So this was till August last year. The numbers have increased since. And if you map it, this is how the map looks. Most of the organ transplants are being done in the southern parts of India, where the organ donation program is more organized. Then most of the transplants are being done in Delhi and in the NCR area. And that's how the program looks right now. We have not tried to, as of now, collect data on the results, but now that we have all the centers on board, we'll now start trying to collect some data on the results now. What we now try to do is we try to create a common sheet on which this data will now be collected. We also now basically have NOTO. NOTO is the central government organization which is involved in organ allocation. So they are also trying to get involved in data collection. And with their involvement, we hope that the data collection will be better and more organized. And we hope that the results will be better now. So that is the status of heart transplant and lung transplant in India. Thank you.
Thank you, Professor Seth. A very nice presentation.
Thank
Now,
you.
over to Dr. Keval Krishnan. He just had an announcement to make before we start the discussions.
So
It's 9 o'clock.
to take this forward, this immunogenetics, I have requested Dr. Uma Kanga and Dr. Ravi Kumar to take a lead and make a one council kind of stuff, so that lot of all the interested people, those who wants to do this, those who are interested in this, they can be part of that council and take it forward, so that we can be the surgeons and the other staff who is involved in transplants and VAD should be more. They should be knowing more about this immunogenetics and related stuff. That's what I want to say. Thank you very much.
Thank you, Dr. Keval, for giving us this opportunity. We will be. I'm really pleased and honored that we will be able to join hands and take this forward. So I will get in touch with the members here, and with the help of Dr. Ravi, I hope we will be able to contribute towards the basic science involved with the transplantation programs. Thanks. Thank you so much. I look forward to more interactions with all of you.
Now, coming down to discussion, any questions from the audience, from the expert panel? So I have one question for Dr. Halloran. So maybe Dr. Jignesh also can answer. Dr. Halloran, you had mentioned that there are some cases, a lot of cases with injury pattern associated with organ dysfunction, especially heart. So you think this is a significant number, or what is it related to? Is it related to primary graft dysfunction or something which happens in the course of the warm ischemic time relating to some fibrosis? So just for Jignesh, Dr. Jignesh also, what is your opinion? So both of you can possibly enlighten us.
I think we have to start thinking about wounding. Every multi-celled organism has the ability to repair wounds, and every organ has the ability to undergo wound repair. Tissues can be stressed, but not wounded. The difference is that wounds have to be repaired. And every time we transplant a heart, a lung, a kidney, it remembers the wounding of the brain death, the implantation, and also rejection episodes leave wounding. So then wounding can be seen over months, and this involves inflammation, dedifferentiation of the parenchymal tissue, matrix remodeling, loss of function, so that we see the loss of the solute carriers, loss of oxidative phosphorylation molecules in the tissue. And all of this is information about how that tissue is handling its damage. And this is universal in transplantation. And once you're wounded, you will never forget the wound. Every organ retains its history of wounding. And what we're trying to do then is understand how that relates to function. A serious wound will leave you with atrophy fibrosis, and that seems to be universal in all the organs. And how this happens and whether we can do anything about this is now one of the big challenges. We can treat rejection, but the organ doesn't forget the wounding of rejection. So we're getting our heads around a new level of information in the tissue, the response to wounding. The cybernetics of the wound may be one of the most profound things to understand in all of disease, because all of disease involves the response to wounding in a tissue. So we're really deeply into that now. And actually that explains dysfunction, that explains GFR in kidneys, FEV1 in lungs, it explains left ventricular ejection fraction, and it explains outcomes.
I think one of the issues regarding transplantation and injury is that there are two components. Every solid organ transplant by nature undergoes ischemia reperfusion injury at the type of engraftment. And subsequently, there's immune-mediated injury as well. And as Dr. Halloran mentioned, how the organ responds to that injury is really what we need to understand more. And what we're seeing here are some of these molecular patterns of those two phenomena.
Dr. Can you hear me?
Yeah,
yes. Yes.
Yeah. Okay. So first of all, I would like to compliment you and congratulate you for organizing this virtual meeting and bringing all the experts together. Thank you very much. Now, as a panelist and as a pathologist, I listen to everything from basics to cell-free DNA to NGS to third generation sequencing and so on, so forth for the diagnosis of rejection. However, we have forgotten that it's a multidisciplinary approach. No single technique can be foolproof. Dr. Jignesh highlighted there is a very high negative predictive value for cell-free DNA, the donor-derived cell-free DNA. I agree with that completely. However, this technique is quite expensive and yet to come to India, right? As far as the diagnosis of rejection is concerned. More than that, I feel that microRNA evaluation in these patients would be much more beneficial because they are actually representing the tissue injury. As Dr. Philip has highlighted, the tissue never forgets this old scar or any rejection which it has undergone in the earlier phase. And maybe in future, who knows, that artificial intelligence is the answer in these patients where we have to monitor the rejection phase, especially in the heart, because heart is less immunogenic as compared to the lungs, or for that matter, kidneys. So lung transplant is much more tricky to monitor. And being in India, I would say that our problem is more of infections than rejection. So we have to control a lot of infection also along with monitoring of rejection in these patients. But indeed, it was a wonderful feast of talks by all the expert speakers, and I really enjoyed the whole sessions. So over to other panelists to give any further comments and all. And it's a teamwork, and we need to collaborate as a team, and multi-center approach needs to be acquired. so that we can have a common platform for the registry, as Dr. Sandeep said, highlighted, more pathologists, more immunologists, and even molecular biologists, besides the transplant surgeons and cardiologists and pulmonologists in the team. Thank you.
I wonder if I might make a, sorry, are you about to go ahead, Dr. Shah?
I was just going to ask, because there's a question from the audience as well, which may kind of builds on what Dr. Nahar was talking about. Is there a program lab within India that is already doing any sort of cell-free DNA assessments of solid organ transplant patients? And I think it would be helpful for, if anybody's aware of that, to really talk about how that's done, what it looks like, what it costs are, et cetera, so that the audience can kind of learn from the Indian experience.
Well, as far as I know, this is not being done in cases of transplant patients. However, the cell-free DNA is being used in tumor patients. So, but it is pretty expensive, and right now it's not coming to the translational value. It is limited to the research presently, at present. Maybe in near future, we are going to have this as much more utility for diagnosing rejection also. But we should not forget that it's all multifactorial. So cell-free DNA alone cannot be used as a marker for rejection. The biopsy is not going anywhere in recent future. I mean, as a pathologist, I can say that.
Along with
Dr. Can we develop it at AIMS as an exploratory thing and then extend it for India?
Sorry?
Dr. Uma, can we develop cell-free DNA at AIMS and then start extending it for the rest of India?
Dr. Uma?
Absolutely. Absolutely, that's not a problem, because I have been doing this, but I am doing it in the tumors, not have started in transplant patients. It's an expensive technique, so but then we need to have a collaborative projects like Dr. Alexander showed, and so many others showed that they are doing a multi-center projects to develop this technique. So that will be for all patients.
I believe that's what's absolutely crucial. And the integrated approach that you had mentioned and which Dr. Lupi emphasized, and his presentation showed just a very immense scale of the collaborative effort that's required to get any of this work done, and how that's going to be crucial in any sort of AI platform, which I agree with you is going to be in future what will direct our sort of diagnostics and clinical advice and algorithms in terms of treating our patients. And I think that calls upon us in India to now form those collegiate and collaborative platforms where we can have a central reference laboratory that can guide us for the sake of efficiency and cost containment. That brings me to two specific questions. On the lines of integrated approach, a question to Jignesh would be, given all the discussions we have had about AlloMap, AlloSure, MRIs, microRNAs, and donor-derived cell-free DNAs, where can the pennies go as opposed to your dollars in terms of units here trying to get a headway on this? And from your presentation, it would appear that the money is probably in the donor-derived cell-free DNA and the molecular microscopy with microRNAs. There seems to be a very significant and I think a really wonderful correlation there. And my second question is directed to Dr. Shah in terms of the microRNAs, which appear to be a plethora of them. But what was nice about the presentation is that it's beginning to show an evolution in terms of separation of certain cohorts of microRNAs into acute cellular rejection and antibody-mediated rejection. And those where there are a mixture are probably for the third type of the mixed rejections. So yesterday we discussed about the differential of the HLA alleles and haplotypes and how they're different distribution amongst ethnic groups. And my question is, how does that relate to microRNAs, and what is the ethnic population sort of race differential there that might go along to further cause confusion with your data? So the first question to Jignesh and second to Dr. Shah.
Yeah, thanks, Kumar. I think the cell-free DNA platform is certainly very promising in terms of making surveillance much easier. Certainly in the United States, we've been moving away from doing surveillance biopsies, and the number of biopsies that we're doing now routinely has gone down significantly. And that also correlates with the lower rates of rejection that we're also seeing as well in our cohort. And I'll certainly defer to Palak regarding the utility of the cell-free DNA platform in being able to distinguish between cellular rejection, maybe antibody-mediated rejection. But I think it's a complementary technology to the biopsy. And I think the biopsy can certainly be improved by the addition of the molecular microscope. So for example, at our center, if we do a 4-cause biopsy, we will always do a molecular microscope with that, because it provides us with valuable information as well. Palak.
Sure. So I think just kind of building on the discussion here, the cell-free DNA assay and the work that has been done really since 2011 in solid organ transplantation, I think, and the clinical utility study which Jignesh presented, I think that really paves the way for using this study, this assay as a clinical non-invasive assessment of allograft rejection. I will say that in our work, we use sequencing, which is extremely expensive. And that's why the sensitivity is much higher, but also the threshold that we use of 0.25 is much higher than what has been used by some of the commercial labs, including CareDx. And so if I think about taking this from a high cost environment and sequencing based, I think you can use certain things like PCR that can be done at a much lower cost and use this type of an assay without the kind of exorbitant cost associated with sequencing. And that could be done in your labs within India, and perhaps in a core lab, at least initially to start with. With respect to the microRNA work, I think it's an additive tool. At this point, we do not have the sheer number of sample sequence and the sheer number of patient sequence as we do with cell-free DNA. And so that although I am very hopeful that we will get much more information about the mechanistic insights for T-cell mediated rejection, antibody mediated rejection, and some reflection in the periphery of what's going on in the graft, I don't think we're there yet, I'll be honest. And that's why I think cell-free DNA, if I'm betting on something right now, it's cell-free DNA, and perhaps in another 5 years or so, we can transform to microRNA, which also will be able to be done with PCR, and potentially can even be done as a point of care test, because it could get down to 10 or 15 microRNAs that you assess locally. I think the last question that you had was around, I guess, maybe HLA differences and racial differences and things like that. And there is clearly a difference in injury levels that we saw in our cell-free DNA paper between Caucasian or European ancestry heart transplant recipients and African ancestry heart transplant recipients. And so I think that that is something that we have to continue to work on to try to understand, why that happens, despite being on the same immunosuppression, managed at the same heart transplant centers. And there's this concept that at least the immunosuppression that we are using may not necessarily be as effective in African ancestry heart transplant recipients. And I'd be curious to hear what the thoughts are about that in Indian or South Asian ancestry heart transplant recipients.
I just wanted to make a comment about that. I mean, one of the things is that the levels of some of the immunosuppression tend to be lower with the same dose. MMF is the drug that comes to mind, so that if you monitor MMF levels and then identify that African Americans have lower levels and you increase the dose, you can compensate for that and address some of the issues of more frequent rejection. I also wanted to mention the impact of COVID on testing, which is during the ongoing pandemic, we really wanted to keep our patients away from the hospital. So we would arrange to do, and a lot of centers would arrange to do COVID testing, testing with AlloMap and drug levels and AlloSure in the patient's home. They go to the home, they draw the bloods, and only if the test indicated potential injury or immune activation
would we bring the patients in to do endomyocardial biopsies? And that's actually become very popular with the patients. And so that may be something, along with telemedicine, that persists once the pandemic is under control.
Maybe I could just comment on, in going back to the MMF studies, I had a role in the MMF studies in kidney, and we did separate out the African American, and the African American really did behave differently. And I was once at a meeting in Cape Town, and Desmond Tutu was there, and he said to the world's leaders who had come to Cape Town, he said, "Welcome home," reminding us that that's where the human arose, and that's where genomic diversity was greatest, and the pressures of the infectious disease burden in Africa have changed that genotype profoundly. Resistance to malaria, etc., sickle cell disease, you know those stories. But that population really is different. And I suspect that my Irish ancestors, my South Asian colleagues have been selected for different things, but the African really is different. And I think that they emerge in the clinical trials that way. The other things are how we would be able to tell us about the socioeconomic burden of the African American as well in the United States. But I think the African genome should really be, we should really recognize its special qualities compared to the rest of us that have been derived from our ancestors there.
I would like to add something here. Dr. Alexander Lupe talked of something called nanostring. That was new to me. So, but even there you need the tissue. You can't just work with the cell-free DNA or the microRNAs there. Maybe that's a new phase to detect early rejection, and that showed quite promising results. So we don't know. I mean, again, we have to have a multidisciplinary and multifactorial approach to tackle this thing in future. And of course, multi-institutional and maybe international collaborative projects would be worthwhile in this aspect.
Yeah, maybe I just could comment. The nanostring is a multi-gene approach. You have to select the genes in advance. It's not a discovery platform, and it's been set up now for paraffin. So there's a way that you can take paraffin tissue, extract the formalin. You've got the disadvantage of formalin covalently changed mRNA, which is permanent, but you still can use that signal in paraffin tissue. So that's the way the nanostring has been set up right now. And you select genes based from another platform, and I think they've got a total of 700 genes they can look at. And then you use what he was describing was formalin fixed tissue goes on the platform.
So we have that platform at AIMS, just recently acquired, and I just wanted to find out from all of you in the US and Canada, the centers, that for us to be able to build up a database at national level, a multicentric and a multidisciplinary approach, what kind of repositories or what kind of samples that you would recommend us to kind of store, so that once we build up a cohort, we would like to either do the microRNA or cell-free or mRNAs or any kind of studies. So we would like to know from you, like Dr. Lupi presented, Dr. Patel, Dr. Shah, that we would seek your guidance in kind of developing the biorepositories which would be used in Indian studies. So it has to be a pan-India program, which we will plan in future, but need your guidance on that. So any comments on that?
Maybe as the one of the senior people, oldest, biorepositories have not been terribly useful. The useful things are to start out by asking what question. You could collect everything, but you'd usually find that you haven't collected it the right way for exactly the question you want. And I see Palak is actually echoing this. You have to know what you want to do with it and what platform you'll be using in order to stabilize that material properly and get the material. So do you want DNA? Do you want microRNA? Do you want messenger RNA? Stabilize it properly for the platform you intend to use. So I think turn it around. What questions do you want to answer? And that will give you the sample and the platform. I don't like biorepositories because too many of them have turned out to be useless because they haven't stabilized the samples for the platforms that have emerged 10 years later.
Oh, I actually used the term biorepository, but what I meant was what specimens to be stored and what time intervals that you actually store the samples. For example, we're doing a study on renal transplants, long non-coding RNA. So we start collecting samples pre-transplant from kidney patients, and then we go on collecting monthly from post-transplant. And we extract, save them in RNA later and do the RNA-seqs and all. So for cardiac and lung, for cardiothoracic transplants, we need to also develop that what would be the useful specimens that all centers should be planning. And we have to have something which, if it's a multidisciplinary and multicentric approach, then all centers should have that capability to be able to handle specimens and storage and time points.
I can tell you that from our experience, we've had a biobank and a blood bank for quite some time, and Dr. Halloran makes an extremely good point that you can't just take tissue and bank it. You've got to know exactly what you're going to be using this data for, and we've been struggling with this. With actually doing a collaborative study with Dr. Shah as well in terms of the type of samples that we have at Cedars-Sinai. We have plenty of samples. The question is, are they useful and are they reliable? And so I think if you're going to start off with setting up a biobank, it's really important to determine what type of samples you want to collect, how you're going to collect them, what sort of aliquots you're going to have for those samples, because you don't want to collect a 10 cc sample of blood that's going to undergo freeze thaw cycles for every extraction. So it does, and it's hard to predict the future. It's what are the future technologies going to allow you to do? So I think it does require a lot of thought in terms of how to set up a biobank and how to store these samples, and it can become quite an expensive venture as well.
I have a practical question. Are you guys participating in the INSHLT registry? You have an enormous number of heart transplants. Have you been submitting your data to the registry? Because if you haven't, I will ask Joe Stellick to reach out to you, because it's really.
No, we have been doing from Chennai for the last 4-5 years. So 2-3 hospitals.
But the rest of the Indian centers.
I think there's probably Delhi escorts, one hospital in Delhi, and there are 3 hospitals in Chennai, 1 in Mumbai. So we have been regularly doing it till last year when there was some break in the software.
Well, I'll mention to Joe that he should reach out to you, and perhaps you can direct him to other people and just try to get all the data, because I think it would be tremendously interesting. And it may be a way of addressing, I think it was Dr. Nahar's question about differences between transplant recipients on the subcontinent and elsewhere.
I asked one question to Dr. Halloran. So you had mentioned in your talk that the graft loss doesn't correlate with your AMR readings on MMDX. Like there are some cases graft loss with AMR diagnosis, there was no graft loss. So how do you explain them? There are some cases with no negative results and graft loss.
Well, thank you for your question. I'm here to describe what we're seeing. I don't necessarily explain what we're seeing. It's a real surprise to us that the way clinicians are creating the environment for heart transplants right now, they're avoiding antibody-mediated rejection of the devastating kind that we used to see. So they're assiduously avoiding the catastrophic situations. And so what we're seeing is a phenotype which does not progress rapidly. Compared to the kidney, what happens in kidney is you start with early onset and then you start to develop double contours in the glomerulus, and that's where the tissue starts to fail, and that's after 2 or 3 years. We don't know what the corresponding weak point in the heart is, and so I'm really asking the pathologists, the cardiologists, how does the heart with stress in the microcirculation fail? Where is the weak point where we can start to see the hearts are turning a corner and starting to go downhill and the parenchyma will deteriorate? Right now, it is a surprise, but I also want to remind you that early onset antibody-mediated rejection is now quite a benign condition for a couple of years in the kidney as well. Microcirculation stress is well tolerated compared to parenchymal stress, which is what T-cell mediated rejection does. And that can be devastating.
I think Alex Lupi has provided us with some very, very useful insight as to what might be going on with antibody-mediated rejection. And our approach to dealing with antibodies at the time of transplant also provides us with some further information. Antibody-mediated rejection is catastrophic at early post-transplant if you have cytotoxic antibodies. And what we've learned from the DUET study using complement inhibitors is that if you can block that early post-transplant, the graft does fine later on post-transplant, even though those antibodies are still there. You've modulated the immune system later on, the graft seems to do fine. What Alex has also shown is that antibodies actually lead to a low level of injury that is often underappreciated on histology for many, many years, leading to late graft failure with allograft vasculopathy. And therefore, I think what we're seeing with antibodies is that it's become a much more benign condition simply because we're much better at treating the acute effects of it at the time of transplantation. And so cellular rejection is now becoming the condition that we really fear when patients present acutely. And I don't know if Alex is still on and wants to make a comment on his study with late failing allografts.
He has possibly has left.
Yeah, he may have left.
Well, I think you make an important point, Jig, that that the effects of antibodies are really delayed and are manifested later in the form of cardiac allograft vasculopathy. And that can be an ongoing later phenomenon, an ongoing source of damage and fibrosis. So I think that's where the significance of antibodies come in. You're right, we can control the early effects. We try to mitigate it by generally not going crossing positive cross matches or desensitizing, a number of other approaches. But I think the ultimate manifestation is CAV.
Can I ask a question of Dr. Kaur if she's still on?
Hello.
Oh, she's still there. Or maybe some of the other panelists, but she had talked a little bit about the C1Q assay for DSA, and this is something that we have not been doing in our HLA lab. We use a number of other techniques to kind of assess the pathogenicity of a DSA. But she also mentioned something around the utility is limited to kind of the first month or something post-transplant. I don't know if anybody else in the panel had some comments or thoughts about that. I hadn't heard that before or kind of learned a little bit about that.
This is about complement fixing antibody?
Complement fixing antibodies with the C1Q esterase assay.
So there's, should I comment on that? Yeah. So it's the C1Q assay is also like the DSA, it's available from 1lambda. It's a commercial assay, and there is controversial data on its utility. So some people say that it's useful early post-transplant, and some people have been monitoring the antibodies even later on. So it depends on the center. You have to develop your own experience on how useful it has been with and usually people combine that C1Q assay with the C3D also. So a combination of the C1Q and C3D binding DSAs, that serves a better purpose. Correlations are better.
There's actually a pretty good correlation between C1Q binding assay and dilutional antibody levels as well. And, we often do a 1 in 8 dilution, and we call it the poor man C1Q. So if cost is an issue, I think dilutional assays are actually they give you a good bang for the buck in terms of what you're looking for.
So do you add on the C3D to it, or just the C1Q and without C1Q?
We only do a C1Q assay. Yeah.
Jignesh, one question to you. You have mentioned that the cell-free DNA, the sensitivity is low in some of those cases. I thought the figure was about 50%. So you think is quite low for practical utility? 50% means you would be missing 50%.
Yeah, really, as with all non-invasive technologies in heart transplantation, it's the high negative predictive value where the test is helpful. There are a lot of things that can raise your cell-free DNA levels, as I mentioned. Tissue injury of any type can certainly lead to that. Infections probably also impact it too. And so the sensitivity is low and variable. And I think that limits our ability to use the test for therapeutic purposes. But it's certainly very helpful in surveillance monitoring, because you can obviate the need for routine biopsies in these patients. The AlloMap is the same. Really, the AlloMap has a very high negative predictive value, but low sensitivity for detection of acute cellular rejection.
Well, there are really two sides of a coin. One is a rule out test, which is the AlloMap, so you can decide when not to do biopsies. And then the cell-free DNA is a rule-in test to decide if you need to do further investigation, whether biopsies to look at AMR or ACR, or else further out coronary angiography.
So just to build on that, right? I think the positive predictive value from the initial studies on AlloMap were 5-6%, depending on what
Correct.
score you use, the patient population. In our cell-free DNA paper, the positive predictive value against endomyocardial biopsy was about 20%, so still pretty low. However, I think if you take those quote unquote false positives, which is what we try to analyze, you can see a number of those false positives precede the development of graft dysfunction, precede the development of overt endomyocardial biopsy proven rejection, and or precede, which we haven't presented, the development of DSA. And so there's a lot of, we're calling it a false positive, and we're saying the positive predictive value of cell-free DNA is low, when in reality, the positive predictive value for things that we would argue are clinically meaningful is actually quite high. But again, we're using an imperfect gold standard, which is the pure histopathologic assessment of an endomyocardial biopsy.
And in fact, in the original cargo study, Mandeep Mehra published a sub-study of it where patients who had these false positive AlloMaps at the time when they were getting blood samples and paired biopsies, and they had negative biopsies, many of these patients would go on and develop either treated rejection or a histologically verified rejection, so that the freedom from treated rejection or 2R rejection was much lower in those patients. So the same sort of thing was seen, that it was predictive. That basically means something is going on that's not wholesome.
So in other words, Dr. Jignesh, if suppose you have a low cell-free DNA, still what would be your approach? Means it still could be a rejection or ongoing rejection or forthcoming rejection.
Well, when you have a low level of cell-free DNA, that's the most comforting test to have. You know that there's nothing going on there, and that's what we look for when we have these results come through. Particularly when you have an elevated AlloMap score, you're always wondering whether that's true or not. The ones which are less clear tend to be the cell-free DNA scores that are slightly elevated and the patient's doing okay. But I think one of the things that Dr. Shah's study has shown is that there appears to be a trend for a rise in cell-free DNA in patients where there may be something going on. And so I think serial analysis of cell-free DNA may in the future become a useful tool in seeing whether there really is something going on or not, particularly in patients who have low levels. And as I mentioned earlier, patients with with antibodies or sensitized patients tend to have a higher basal level of cell-free DNA, at least on the platform that was used by CareDx. And I think the analysis of the result of the test that you're looking at, you have to look at the patient as well. Is the patient sensitized or not? Has the patient had recent rejection or not, or what the patient's rejection risk is, and then you kind of use that information. As mentioned earlier, it's not a test that you can use in isolation. You have to integrate all your data and come up with a plan for further diagnostics or therapeutics.
And then what would be your advice? For example, you have all these technologies, MMDX, EM biopsy, small messenger RNA, cell-free DNA. So how would you use all this as a clinician, just as a message to audience?
Well, our first goal is to reduce the number of surveillance biopsies we need to do. So if we can use non-invasive strategies to rule out the need for biopsies, that's great. So, for example, the AlloMap has been very useful in reducing the number of biopsies we need to do in patients who are not sensitized. I think the cell-free DNA platform will provide us with additional level of comfort in patients who are sensitized, where the AlloMap test has not been useful, and in reducing the number of biopsies we need to do there. Now, if any of these tests are positive and you're pushed towards doing a biopsy, I think it's useful to consider adding a molecular microscope to that biopsy to get further information.
Maybe I could just make a comment. In relationship to platforms, there's granularity in each of these phenomena, and the platforms are evolving. So I really liked the recent Circulation paper because I believe they're using shotgun sequencing rather than highly multiplexed PCR. And I think actually they've found things that are going to be very important. I think that the granularity in the measures that you're using will evolve. Right now, using percent donor-derived DNA is not a good idea. You've got an equation in two unknowns, and we all know that. And both of the unknowns can move. And this is not the way you want to do a clinical test. You want to have an absolute number, but also probably the length of the fragments. So the same thing with DSA. There's enormous detail in DSA, which really we're not making use of the potential of getting the DSA and turning it into a test which says this DSA is highly likely to be pathogenic and this one is something you can sleep easily about. So let's look more at evolution of the platforms and the potential granularity of the platform. And for India, again, making choices that prepare samples for the evolving platforms would be very important. And including small RNAs, large RNAs, DNA, et cetera. But think about where the platforms will be in 5 years. Listen, there's a serious lesson here. We made some platform mistakes and found ourselves with an orphaned platform that wasn't going to be technically supported, and then spent a large amount of money and had to abandon everything because we couldn't use the results. So that is actually a very important lesson. Think about where the platform will be in 5 years and try to anticipate it. And good luck.
One question, Dr. Palak. I mean, you had some studies about the ethnic kind of background and rejection. But there have been a lot of studies from India, especially. I don't know whether you have been aware. There's one large study from Harvard and multiple groups, Dr. Reek. So they had their paper was on origin of ancient Indians and relation to modern population. And they actually shown that the Indian DNA is actually mixed like mixed DNA, basically. You have ancient North Indian, ancient South Indian, everything is mixed up. So what do you think would be, like, suppose you had Indian donor, Indian recipient, you think it would be different from, or would it be easier here? We have not actually seen our data for overall rejection in detail.
Sure. So I think the concept that we're getting at is that the HLA region, at least if we focus on that for transplantation, is highly polymorphic. And we look at it, at least in solid organ transplantation, at a very superficial level. And obviously our bone marrow colleagues look at it in much greater detail. And if you look at certain populations, and the populations that I'm most familiar with is the African population, is that even if you match at an HLA level, at a major allele level, you can see that there's a lot of polymorphism below those HLA with the smaller alleles. And that seems to contribute to the level of injury, such that if you match an African-American organ transplantation organ with an African American, you still end up seeing higher levels of graft injury than you do if it's a better immunologic match. And there's some data that we have started looking at in our cell-free DNA data, because we can look at all of the SNPs and come up with and focus on the HLA region. I have not looked at South Asian ancestry because in large part, South Asians are actually underrepresented in large genome-wide association studies, right? So if you look at these large analyses that have been done really across the globe, we can find patient populations from clearly European ancestry, African ancestry, even our East Asian colleagues. There's actually large nationwide genotyping programs that have been done. But I think that's something that's important to do for South Asians as well. And so, back to Uma's question earlier, I think if you are collecting bloods at all your transplant centers and things like that, it is important to be thinking about collecting DNA so that we understand what the genetic architecture of Indians or South Asians is much better than we do currently.
We are actually doing that in some parts, some populations, we're able to do it. And we're also storing all the DNAs for. But that's also is only at the moment as a research program. And we have started doing the NGS-based HLA typing. But at times, I've heard from some transplant program, that what they do is that we have high-resolution HLA data submitted to allelefrequency.net program. So people do try and extrapolate information that if this is a population reported, what would be the high-resolution alleles most likely be? And accordingly, predict the DSA as to what it is. Is it really likely to be a donor-specific antibody or not? So in those predictions, people are doing. But for matching, definitely we will have to actually do the recipient donor and only then have the information. So we're trying to build up that database in Indian population. It will take time till we have, huge numbers tested at high resolution. But that's the goal, that we should have that information available.
Then what the studies in Winnipeg and other studies now have suggested that if you can anticipate the populations at risk for under immunosuppression non-adherence, for example, the young male with a pickup truck in Canada, very high rate of non-adherence, then you should probably emphasize the anticipation of epaulets or epitopes, which are going to generate DSA. So a combination of thinking about the risks in the population and how to mitigate risks in special circumstances, the adolescent girl, the underinsured patient, which is common in India, that sort of thing, and then try to prevent them from having the epitopes which are likely to generate DSAs. It's a gigantic problem, but it's all solvable ultimately. Actually, I also think we should incorporate structural information, and sooner or later we will understand the HLA and the antibody interactions in a three-dimensional structural way and understand what are the how does a determinant create a hexamer of IgG that will activate effector mechanisms. And that would actually really inform how we allocate organs.
So I touched upon this yesterday in my talk, but I couldn't allude it in detail, that we should be looking at the epitopes, eplets, and look at the acceptable mismatches using the HLA matchmaker, or also use the PERCH algorithms developed by the Leiden University centers. And they are able to predict the alloimmune response that's going to be generated. And because we don't have a huge cadaver donor program here in India, and organ allocation is not going through all of these investigations, once we develop these softwares and we have a huge deceased donor program, then definitely these predictions are going to be useful. And what you're talking of, the structural, people are also started measuring the electrostatic potential, of the HLA molecules, and using that, trying to predict whether they are going to generate DSA or not. So a lot of things coming up, but we will have to incorporate in our studies and see the translational value of those. Very nice question, Dr. Hilleron. Yeah.
Yeah, it'll be important to leverage the open source platforms that Alex Lupi is developing, and that'll be a very useful resource in the future for us to integrate the information that we need for this complex area.
Absolutely.
Dr. Jignesh, there was one question about microchimerism and whether it interferes with the cell-free DNA estimation. There was a question from the audience.
I don't know the answer to that. Maybe Palak could provide some greater insights into microchimerism and DSA. Intuitively, I think that there may be something there, but as far as I know, there's no data that's available regarding that.
We've been trying to look at this in greater detail, but I feel like we're still very early to try to truly make some sort of meaningful, kind of conclusions from the analyses. But certainly looking at this as well. I have a quick question. Does each center in India use a different immunosuppression protocol, different monitoring strategy, things like that? Because in America, I would say of the 130 heart transplant centers, everybody does something very differently. And honestly, I think, I think Alexander, he talked a little bit about this, right? We need to standardize our thinking and our processes for monitoring patients. But I also wonder if we don't standardize how we manage patients after transplant, whether there's just so much heterogeneity. So I'm just curious if you guys are all kind of trying to do the same thing, at least with terms of drug therapy and things like that, or is everything kind of center specific?
Indian Society of Heart Lung Transplant with ISCTS, we had a standardized recommendation. We had put it in our website consensus. So here, about 90% of centers use induction with basiliximab. And then we use low dose steroid, which comes down to about 5 to 10 milligram at 6 months. So most of the centers now are following it. And this was actually, we had done a consensus 2 years back and put it. So most of the centers use the same. They use the induction with basiliximab and low dose steroid after 6 months. And my MMF and the CNI would be the same, like tacrolimus.
So Dr. Ravi, are the rejection rates similar across?
We are not analyzed, madam, exact data. Although the Dr. Balakrishnan in our publication, we have a rejection which is less than 10% overall the first 1 year.
And is that a global phenomena similar globally?
I think it is less in you, Dr. Jignesh, possibly.
In general, the rejection rates in the US are in the 5 to 8% range. I think Palak's recent study showed the cell-free DNA cohort, the rejection rate was around 5%, if I recall correctly.
But our infection is a significant problem, madam. We have about 5 years now, we observed we have 450 cases. We have lost so many patients with fungal, like mucormycosis, aspergillosis, tuberculosis, and some of them with CMV, systemic cytomegalovirus. The infection is significant. And possibly that's why we have to tailor the immunosuppression. We are not able to get a right answer.
So unfortunately, we are in the region with a lot of pathogen load. So we have to handle that, and hopefully some more information will come in.
Yes.
I think beyond immunosuppression, the issue of prophylaxis for opportunistic infections is a really important area that needs to be focused on, and it's a regional issue. Even the prophylaxis that we use in Southern California is different to what's used on the East Coast because of things like valley fever and cocci, which is endemic in our area. We see more surveillance for Chagas, for example, it's more prevalent. So I think it's a regional issue, and I think that it's really important area to focus on. Other than we try and minimize immunosuppression as much as we can, but I think there are additional areas where we need to focus, particularly with regard to prophylactic regimens.
So I'll mention one thing. We didn't get to cover this during this meeting, but something to definitely look up, and I think there's a lot of excitement around this, is that one of the things you can do with the cell-free DNA data, especially if you're doing sequencing, is you can kind of look at the viral and bacterial and fungal kind of landscape of microbes that are circulating in the bloodstream. And there's been a lot of work that's been done around anellovirus and toll-viruses that suggest that that may be a marker of not only rejection, but also about the overall degree of immunosuppression. And I think some of the labs are using this data to say, well, as opposed to doing a CMV PCR or EBV PCR or something like that, can we use this cell-free DNA data, not just to quantitate the amount of injury do we have, but also to diagnose or infection ahead of time. And so I think that's an interesting area, because I think we, again, like I said during my talk, we have to kind of move beyond just rejection and also get towards these other complications that are plaguing our patients.
Palak, there was one study using this stock tenovirus quantification. So you think, I mean, these things, I think is in the research realm. They're not yet come for validated kind of commercial application. So you would suggest Silex or something like that?
Lots of different ways that you can analyze it. And there's been no standardization yet about what's the best way to do that. But that's exactly the technology that we'll get to over or under immunosuppression.
Dr. Kaur, if he is there, because Lalpath lab, I mean, they are developing the cell-free DNA and MMDX. They say that they're going to develop both. I don't know whether, Madam, if you're there.
So I could answer that. We've already validated the cell-free DNA, like I said yesterday, and I think we'll be ready to roll it out for clinical use by the first week of February. So cell-free DNA is very much on the way. The MMDX, we are in talks with Dr. Helleran, and I think the COVID delayed it a bit, but I think we take that on after we started the cell-free DNA. We have the equipment in place, and it was all about laying out the lab and things like that. So I don't think we are very much far behind, but yes, we will catch up. We will look at that maybe about 2 or 3 months down the line for MMTX. But as of now, it's just a cell-free DNA.
What's your advice, Dr. Jignesh and Palak, as a clinician? So what should I do? Like I have MMDX, I have only cell-free DNA, I EM biopsy. So I don't have this other one, AlloMap. So how do you think? I mean, is it okay, or I mean, or is there a pitfalls in this? What is your advice?
I'll let you go first.
Well, I thought, Palak, it might be a good platform to push cell-free DNA for you. I think for India, you've got to really look at what is affordable for the patient and what's going to provide you with the greatest degree of safety for the patient. And I think the cell-free DNA platform certainly seems to be the most promising in terms of being able to provide that level of surveillance at the lowest potential cost. Of course, when the patient gets into trouble, you're going to have to do a biopsy, and at that point, you may want to consider doing MMDX when it's available there to provide you with the additional information.
So I've been obviously thinking about this a lot, and I think having you guys talk a little bit about some of the constraints, I think makes it come up with a more practical approach to this, right? And so if I had to outline very specifically what I would like to be seen done, and hopefully we can do this as part of a trial, I think there's a role for a biopsy and hemodynamics that you get at the time of a biopsy as well. Weekly for that first 4 weeks when there's just a lot of things happening with the patient, aside from just immune monitoring and surveillance and rejection. After that 28-day period, I think then a non-invasive monitoring strategy with cell-free DNA at whatever monthly intervals. And when that cell-free DNA measure is elevated, at that point, then that should trigger things like donor-specific antibody testing if available, endomyocardial biopsy if available, plus or minus a molecular microscope, plus or minus an echocardiogram, at least for heart transplantation. That's kind of how I'm thinking about the future of using this as a non-invasive monitoring strategy.
I think that's
the
really
first
important.
biopsy should have MMDX. For example, our hospital, we can afford, I mean, our patients can afford, whatever Jasmeet Lalpat would offer. So you think the first biopsy we should do MMDX, or we should wait for some kind of event?
I think Palak has a great outline there. The cell-free DNA is not very reliable in the first month or so because the levels are intrinsically high. Alamap's certainly not validated for the first 55 days. I think our purpose for doing endomyocardial biopsies in the first 4 weeks is really to pick up those early cases of rejection that we do see, and also look at the hemodynamic assessment as well within that first month. And in the US, we do weekly biopsies in the first month, but I think in practical terms, you could probably cut that down to 2 biopsies in the first month.
Yeah.
And
so we do a biopsy routinely at 3rd week here. So
Yeah.
you think you should do MMDX also as well in the first instance, or wait for?
Yeah, I don't think necessarily you need to do the MMDX for that surveillance biopsy initially. Again, it's a cost issue. It's nice to have it there, but certainly wouldn't do it. Now, the question is, should you save a sample in RNA later? And if your patient is in trouble, send that off. That's certainly something that can be considered, but there are timing issues for that platform.
Maybe I just comment, the MMDX, we're finding a lot of people use it in the problem biopsy. A pristine biopsy is very likely to be molecularly normal, although some of them do have low-level antibody-mediated rejection in particular. But the problem biopsies, where there's a lot going on, it is very hard to dissect multiple diseases. And that's where the molecular microscope really, I think, is helpful.
So I have a question for Dr. Patel. I just want to know what is the kind of gap you give for a heart biopsy before you actually go out and look at the cell-free DNA? Do you wait just 24 hours, or do you give it a little longer time in practice in your centers?
You mean doing a cell-free DNA at the time of a heart?
To a biopsy, yes.
Typically you always do the cell-free DNA before the biopsy if you're going to do it concurrently, because the endomyocardial biopsy will increase your circulating donor-derived fraction. So if you're going to do it concomitantly, then in the studies, that's how it was done prior to the biopsy.
And should you feel the need to do it after you've done the biopsy, how much gap would you recommend?
Well, I wouldn't routinely do it necessarily, because if your biopsy is negative and the patient's doing well, I'm not sure that you need to do a cell-free DNA assay soon after the biopsy at all. And so I think I can't see a scenario where you do a biopsy and you're going to need to do a cell-free DNA within 24, 48 hours after that.
It's actually the opposite. The opposite. The purpose of the cell-free DNA is to determine if there's injury and do you need to investigate further. And so that's why you do it. You don't do it as a follow-up for biopsy. You're doing it to look for injury, and then you do the biopsy to see what type of injury there is. Or if it's later on, you do coronary angiography if you suspect CAV.
If you're in a situation, just to speak, the half-life of the assay is about 16 minutes or so. Cell-free DNA is cleared very quickly. And so, if you're doing it, you could probably do it the day after a biopsy for some reason if you wanted to, but certainly would not recommend doing it the same day as you have already done a biopsy.
But clinically, there's no reason to do it if you have the biopsy results. I guess the other thing to note is that there are periods immediately after transplant where you can't use either technique. Some of the genes in the AlloMap are very sensitive to and very much affected by higher doses of steroids, which means greater than 20 milligrams of prednisone a day. So, and in the original cargo study and the follow-up cargo study, assay samples were not obtained within the first 55 days. So we don't know what to make of samples within the first 2 months in essence. So we don't get Allomaps within the first 2 months. We can get donor-derived cell-free DNA starting, I guess, from about 2 weeks out, once you've finished the washout period of ischemia reperfusion. So if about 2 weeks out post-transplant and beyond, you can do you can use donor-derived cell-free DNA as a screen.
Yeah. So I just wanted a little input on how frequently are you recommending? I know the recommendations, and I spoke about it, is every month for the first 1 year, et cetera, et cetera. But if we were going to plan a strategy of implementing the test in India, and we can do it so frequently with our patients because of cost, et cetera, what would be a minimum requirement of the cell-free DNA that you would suggest that we could, we could start with, rather than want it every month, or
Depends what your goal is. It depends what your goal is. If your goal is to minimize biopsies, then you fit it into the biopsy schedule. So if you're biopsying, I think Ravi mentioned 3 weeks out, you do it 3 weeks out and see if you need a biopsy or not. I think that's the way we've been doing it. And as I'd mentioned, with the COVID pandemic and the goal being to minimize patient travels to the hospital to potentially get exposed to COVID, we would assay donor-derived cell-free DNA and Allomap at the times that we'd ordinarily be getting biopsies. So they fit into our biopsy schedule, and then would base the determination of whether to get a biopsy on the results of those tests.
So launching this kind of home collection for cell-free DNA is not an issue in India. There's enough number of labs who are, even before the pandemic, have been doing a lot of home collection. So that's a system which is already very well established in India for many years. And so I don't think it's about getting the patient to the hospital. It's just about, I think in our case, it's going to be more about the cost of the test, and which is going to actually, along with the clinical requirement, going to guide how frequently we perform the test.
Right.
So I think we have to just learn from our experience, vary from patient to patient, and things like that.
So the INSHLT recommendations for surveillance really is a tapered protocol for biopsies, where we do up to weekly biopsies in the first month, and then every other week for the next 2 months, going to monthly up to the 6th month, and then every other month up to the 1 year. Now you could essentially do the biopsies for that first month and then replace the rest with cell-free DNA monitoring. And you can also then reduce those numbers, the number of monitoring episodes that you need to do for that, depending upon the immunological risk of the patient and whether the patient can afford the testing or not. And I think you could certainly come up with a protocol that has been modified to allow you to do that. But the important thing is that if you're doing the monitoring, it's the first 6 months that you're going to have the biggest bang for the buck in terms of your rejection risk. And then after that, you can comfortably really reduce the frequency of monitoring in stable.
One more last question. How do you place cell-free DNA if it were to parallel the DSA? I give you my situation. If I were to give a patient a choice, cell-free DNA or DSA or both, and if they say one, how would you place them against each other?
So DSA monitoring really doesn't have to be as frequent as rejection monitoring, particularly in the first year. Typically, we monitor DSA at 1 month, and then 3, 6 months, and then at 1 year. But you could certainly make an argument for monitoring much less frequently. particularly in patients who are not pre-sensitized. So I think the DSA monitoring can be much less frequent than cell-free DNA monitoring in that first year.
The major
If I could just
concern
comment.
it's the economy that the patients being able to pay for the investigations. Even pre-transplant, the waiting list patients cannot afford to go for the DSA monitoring routinely, the single antigen testing. So especially in the government center, then it's unlike US when everything is covered by the health insurance. So it's very difficult to even do the pre-transplant monitoring. And the same is going to apply to our post-transplant, that we'll have to be very, it's going to be a difficult choice for the clinicians, for the laboratories, and for the patients, all of us, to be able to decide. Really, the cost is a big issue. But yeah, if the numbers go higher and we are able to get a better prices, then definitely we would be able to apply to our centers.
The comments. One, Dr. Jignesh, since all these questions are coming, is INSHLT coming up with some committee for new guidelines or consensus document to integrate all this, what we are talking about?
Yes, there's actually been an ongoing
yes, sir.
Multiple task forces which have revised the 2010 guidelines. It'll be incorporating all of this. And I think, Jig, you're on one of those
Yes.
task forces.
Yes, the 2010 guidelines are currently undergoing review. The update is currently undergoing review, and hopefully we'll be able to post it. We're actually looking into the possibility of having it as a live document that can be changed rather than doing it every 10 years. Updating that guideline as we go along. And that's certainly something that may be much more useful than having a document that ages very rapidly.
Any other comments from any expert panelists?
Maybe I could just make two comments. One is people talk about cell-free DNA as measuring injury. When we've looked at kidney injury, we haven't done this yet with heart, we're going to, but in kidney injury, it's not a particularly good measure of kidney injury. It really is quite specific for the microcirculation injury. So if you look at all the biopsies we have in the first 200, and you say what molecules are associated with cell-free DNA release, what you come back is the antibody-mediated rejection molecules. And then you take out the rejection biopsies, and then you look at the injury, and it doesn't seem to correlate particularly well with the injury molecules. And in the hearts, or in the kidneys at any rate, that have atrophy fibrosis, which is a very serious phenotype, they don't release cell-free DNA consistently at all. So when you start to think about injury and in relation to cell-free DNA, you have to be critical and try to find out what type of injury might this be if it isn't rejection. And the second point is, we're finding a fair number of hearts and kidneys with a solid antibody-mediated rejection phenotype that don't have any DSA. Now, it's not uncommon. The majority do. And of course, we find lots of DSAs that seem completely innocent and are producing no phenotype at all in the biopsy. So when you're thinking about these platform outputs, you have to take a lot of things into account.
Comment, Dr. Jignesh, because there's a clinico-pathological kind of correlation. So where do they meet? Anything else you want?
Yeah, I think it's a complex picture, and at the end of the day, you have to look at the patient as well. And what does the patient look like? What does the graft function look like clinically? And really have to what are the hemodynamics, and integrate all that data with what you're seeing coming from the lab as well. And I think that's where the art of management really comes in. There's no single test that's going to help you. There's no single clinical parameter that's helpful. It's a question of looking at the overall picture and making a determination. And I think it's important to not lose focus on that.
And what we tell trainees, whether it's these tests or hemodynamics, it's what does a patient look like? What did the patient tell you? So it all comes down to clinical judgment and incorporating the tests into clinical judgment.
So anything else? Anybody else like to add anything? Dr. Palak?
I want to congratulate all of you on just a fabulous meeting, really superb, and all of your success in building this transplantation in VADS in India, and we'll make sure it's not such a secret anymore, and let the secret get out if that's okay.
Thanks to Dr. Ravi for organizing this.
Thank you.
For
Thank you. Thank you. Thank you so much.
Thank you all.
Thank you, Dr.
Very
Ravi
wonderful
Kumar.
session. We learned a lot.
Thank
look
you.
forward to collaborating.
Yeah, we'll see you virtually at the INSHLT.
Yeah, yes.
Hopefully.
We look forward to a good collaboration. Thank you.
Thank you,
Thank
Dr.
you.
Ignesh. Thank you, Professor Halloran. Thanks. Okay.
Thank
Bye-bye. Thank you.
Thank you, Dr. Umar.
Thank you.
Dr. Jasmeet, thank you.
Thank you. Thank you, Dr. Ravi.